that I'm still allowed to go to the office. Whatever, it is what it is. We develop some resilience, and
exactly.
as long as our dear ones are healthy and nothing happens to them. Yeah. All right, I think it's almost 4, right? Oops. Are you still there, Ute? Hello. Oh, no.
My connection is bad. Can you still hear me?
Yeah, yes, you're back now. That's good.
Okay, hopefully my connection.
So. It's a pity. Hello.
Okay, I think Ute's connection is back. Okay, she rejoined.
Okay, can you still hear me?
Yes, I can hear you.
Okay, I tried once again to welcome you all, and I'm really happy that so many of you joined us for this keynote lecture on the social neuroscience of empathy. And since I'm afraid this network connection might be unstable for the next few minutes, I try to be fast and let Klaus take over. I would just like to shortly introduce him. Klaus Lamm is the full professor of biological psychology at the Department of Psychology in Vienna. And he's member of the Academy of Science, of the Austrian Academy of Science. He studied psychology, and he also did his PhD in Vienna, and then he spent some time abroad in France. He did a postdoc in Switzerland, and he was research associate at Decity's lab in Chicago, and then came back 2010 to Vienna, where he took over this professorship. His research focuses on the neural underpinnings of empathy and prosocial behavior. And there he uses multimodal imaging, EEG and fMRI, but also psychopharmacology and psychoneuroendocrinology. And also, as I heard recently, comparative approaches in ravens and dogs to compare those processes in humans with animals. And I'm thankful that he took over this opportunity to present his research to you, and I would like to hand over the floor to Klaus. Thank you, Klaus, for accepting the invitation.
All right, thank you, Ute. It's really a pleasure to participate in this exciting new format. I'll just jump right in because time is short. We have 30 minutes, and I want to give you an overview of what I will talk about in those next 30 minutes or so. I structured my talk into 2 parts. In part 1, I will focus on empathy and affect sharing, and in the second one on empathy and self-other distinction. And those are 2 important components of empathy that we have researched on in the last 10 or 15 years, we in the field of social neuroscience. So let's start with part 1. One exciting finding, more or less 14 to 15 years ago now, was that there seems to be something like an activation sharing when people are in pain themselves, or when they observe, for example, pictures of others being in pain, or pain being inflicted, as you see it here, with an electrode and a pain stimulator, an electrical pain stimulator. So what you see in this network over here in green is the activation when people are in pain themselves, and in red you see those parts of the brain that light up using fMRI conjointly when someone observes someone else being in pain, or when someone is in pain, him or herself. And this so-called shared activation model was used then to basically make claims about being in an empathic state or empathizing with others means that we kind of reactivate similar bodily or emotional representations. So basically, based on our first-hand emotions or based on our first-hand neural and cognitive responses, we are able to share the affect, or in this case, the pain of another person. However, there is one problem, or I mean, there are a couple of problems, but one that we want to focus on today, and they are as such. So activations, as we measure them with fMRI especially, but also with other methods, are not one-to-one mappings of representation. So in a way, the language of the brain is not necessarily the language of cognition or functional processes. The second problem is that fMRI is a neurocorrelate measure, so we don't get any mechanistic insights. We might as well say that, okay, there is an activation in an area that is also active when I'm in pain, when I'm observing someone in pain, but how that activation relates to then understanding the other's plight, that is unclear. So the idea we had, or the research program we started a couple of years ago, is to gain deeper insights into something that I would want to call here the functional correspondence, or the level of representations between first-hand pain and empathy for pain. Where does this idea or this concept come from? Some of you may remember in the 90s, there was a very similar discussion in the field of visual imagery, or imagery in general. So one citation here, the functional equivalence hypothesis, the proposition that imagery, although it does not result from stimulation of sense organs, is essentially the same as perception in the way that it functions. Complicated sentence, but basically the idea we borrowed from there. So we wanted to look at the functional equivalence or correspondence between being in pain oneself and empathizing with the pain of another person. How did we do that? With a actually rather simple approach, if you think about it more deeply. The assumption, or the main pillar, so to say, on which this research program builds is the following. If it's really the case that first-hand pain representations are in a way used or essential to empathize with the pain of another person, so indicated by this arrow here, then shouldn't it be also the case that if I systematically reduce, for example, or also increase my own pain experiences, that there should be a carryover effect to the empathy for pain. In other words, if I give people, for example, a painkiller to help them deal with their own pain better, then this should also reduce empathy for the pain of another person. So this is the question mark, the thing we wanted to do. We, however, did not give them a painkiller, but we used the placebo analgesia manipulation. I will get into this in a second or so. Here, I will report a multi-method approach. So we combined several methods to go at this, to basically test this essential hypothesis. If I reduce people's own pain experience, will that also affect or reduce, more specifically, their empathy for pain? The basic design, or the basic experimental approach we used here was that we always used electrical painful and non-painful control stimulation. And we always had 4 conditions, or a 2x2 factorial design. So we exposed participants, for example, in the scanner or in an EEG lab to these painful stimuli, either to themselves or to another person, and the control stimuli. So this is the basic design. There was always a joint participation, meaning that there were always 2 people interacting, in a sense that one person was, for example, lying in the scanner and the other person was outside. Also part of the design, and this is important, and that's why I mentioned it here explicitly, is that we always ask for 3 types of self-report rating. So this is basically the subjective component of our paradigm, asking people how much pain they experience, how much pain they think the other person experienced. So this is more a cognitive evaluation of how painful a certain stimulus was for the second person. And unpleasantness, that is basically asking participants, how unpleasant, in a way, painful was it for you to experience the pain of the other person? So when the other person got a painful shock, how much unpleasant emotion did that create in yourself? We had a between subjects design in these studies. So we compared a placebo analgesia group versus a control group. Gender, age, education, and so on were more or less kept equal. Those are the classical undergraduate student guinea pigs of many of the experiments we are doing these days in cognitive neuroscience and in social neuroscience. What does the placebo analgesia group mean? So basically, in one of the 2 groups, we gave people a placebo painkiller, so basically a a pill without any active psychopharmacological compound. And this pill was told to the participant is a potent and very powerful painkiller. On top of that belief induction that is well established in the literature, we also used a learning trial procedure to reinforce this belief that what participants were just given is really indeed a painkiller. I can go into the details of that procedure if you want in the discussion. But basically, we followed standard procedures developed in the literature for the investigation of pain analgesia or pain placebo analgesia in the pain literature, so not in the empathy literature. Let's go to the first study. So this was planned more or less as a type of pilot study, but turned out to produce very interesting results in line with our hypotheses. So it was an ERP study, event-related potentials. Basically, if you can see it up here, participants were fitted a cap with 64 electrodes that were measuring their electroencephalogram. And from that, we derived one event-related potential component, the so-called pain-related P2. The pain-related P2 is a deflection over the frontocentral area, so that would be pretty much here. So there are electrodes there that pick up this component. And the interesting part for us was that this component is sensitive to placebo analgesia. So it shows modulation when you do placebo analgesia experiments without the empathy component, and it has been source localized to parts of that pain matrix or that shared activations matrix that we have seen before. So to cut it short, you can consider it as an ERP signature of the affective motivational component of pain. There is some discussion on whether that's really the most precise description, but for the purpose of our talk here or in our discussion here, let's say that's enough. So let's jump right into the behavioral findings. So these are self-report ratings. And remember, I told you there are these three different types of ratings. There's a self-directed pain rating. So how much pain am I experiencing when I'm giving a painful shock? And I'm subtracting that self-report to the non-painful control stimulation. How much pain do I attribute to the other participant, to the second person that I'm supposed to show empathy for? And how unpleasant was it? Again, pain minus no pain. So how unpleasant is it when I know the other person gets a painful shock compared to a non-painful control stimulation? So it's important that we plot here this difference between pain and no pain. So unspecific effects are always subtracted. What you see here then is the control group. So the group that was not undergoing the placebo analgesia manipulation. And here in light gray, the placebo analgesia group. This is interesting in itself, but it's nothing more than a manipulation check in the sense that our placebo analgesia manipulation that we used worked out well because participants in the placebo group reported analgesic effects in the sense that they self-reported to experience less pain than the control. What's interesting for the current purposes is how does this now affect how people empathize with the other person? And this is a very nice finding in line with our hypothesis. They attribute less pain to the other person, and they also experience less negative affect or less unpleasantness. So that was more or less in line with our hypothesis. And what's also interesting, and this is probably was not predicted in that all that specificity, was that the effect sizes in the domain of self-experienced pain and empathy for pain were not distinguishable. So the amount of statistical effect, so to say, was the same for placebo analgesia working on my own pain versus on rating the pain of another person. On the level of the brain, this is this P2 component that I talked about to you before. We now see plotted the distribution. You can see it's slightly posterior, maybe than the central part. Or the frontocentral part is more or less smack on the CZ electrode. You compare here the control with the placebo group, and you can see a reduction in amplitude that is quite substantial. So this is what has been described in the literature before, the sensitivity to placebo analgesia when painful stimuli are delivered to the self, to the participant. And the interesting thing is here that we also see such a reduction in the amplitude when the other person, the second participant, is getting the painful shocks. You will also notice that, of course, the amplitude is much lower. So that simply reflects that getting a painful shock yourself versus observing it and responding to that in your brain results in a lower amplitude. But in terms of the amplitude differences, this is what is plotted here. Again, you don't see any difference. So no significant difference in terms of the effect size. And the effects are also specific because we don't get the same manipulation or modulation for the non-painful control stimuli. So that was great, but those of you who have worked maybe with EEG know that its spatial specificity or its spatial resolution is rather limited. We also had the discussion of what the P2 really measures. So we did a follow-up study with a much larger sample. And here we used fMRI with pretty much the same design, an alternation of getting painful shocks oneself or observing indirectly another person who was getting such painful shocks. We focused on so-called region of interest analysis from the left anterior insula, the right anterior insula, and the MCC. So those are three of the nodes or of the areas that you have seen in the slide at the outset of my talk, where I talked about shared activations. I'm jumping right into the behavior again, so the self-report. This is now a slide that looks slightly familiar to you already. We replicate what we have found before, so the control group and the placebo group in terms of a manipulation check. Placebo analgesia, a reduced self-report of pain for the self-directed pain ratings. But this is more interesting now. We see exactly the same pattern as in the ERP study. Slightly different ratings here, but in terms of the difference, nothing much changes. So again, in terms of the effect size, how the placebo analgesia acts on the modulation of self-experienced pain and empathy for pain, there is no statistical difference. So that replicated the ERP study findings in a much larger sample and in the kind of hostile environment of an MRI scanner. When we look at the level of the brain, so the neural responses in the three regions of interest that we were analyzing, I'm first plotting again the self-directed pain. So this is the modulation of activation you see when participants are receiving painful shocks themselves compared to non-painful control stimulation. You see a nice modulation in all three regions of interest. And you see the same pattern, but again, with a reduced amplitude, but not a reduced difference for the left anterior insula, the MCC, and by trend also for the right anterior insula. So you can say that's great, but now how do we really get above kind of this limitation? How do we get over that? That both ERP and fMRI are just neural correlate measures. So we cannot really say something about the specific neurochemical processes or the causal mechanisms involved in generating these findings. The way we tried to get at that was by running a psychopharmacological experiment. And here we used the opioid antagonist naltrexone. We now had a slight change in the experimental design. We have now, again, two groups, but they differ from the ones before in the following way. In both groups, we induced placebo analgesia. But in one group, after placebo analgesia was induced, we also gave them naltrexone, the opioid antagonist naltrexone. And the other group did not get that. So they got another placebo without any psychopharmacological or pharmacological compound. And the idea behind this was again borrowed from the literature on placebo analgesia, so from the pain literature, so to say. So there, this procedure, either using naltrexone or naloxone, which is injected, is to test whether an effect of the placebo analgesia is mainly mediated by the opioid system. And if this is the case, there should be a difference between those two groups. And that's basically the main hypothesis we were testing with this third experiment. In the line of experiments I'm presenting today, if the effects disappear with naltrexone, they are related most likely to an opioidergic mechanism. Here we only have the behavioral findings, and you can already see a stark difference to the plots from before. So now we have the placebo naltrexone group. So that's the group where placebo analgesia was induced, and then they were given an opioid antagonist to block the opioidergic effects. And in the self-report of self-directed pain, so pain they were experiencing themselves, you see a reversal. So there is no more analgesia, but hyperalgesia. So they experienced more pain compared to the placebo group that was not given the opioid antagonist. And you see exactly the same pattern for the empathy for pain ratings and the unpleasantness rate. So to sum it up, next study, sorry, I forgot. We also have that one since more recently. You can ask yourself now, okay, that's great, but that's only a behavioral study. There can be many things that can explain that. Do we also find this on the neural level? So far, we only have ERP experiments. The interesting thing, and that's very comforting, so to say, or very reassuring, you can replicate the behavioral results with an independent sample of the psychopharmacological behavior-only experiment. So that's a thumbs up, so to say. In times of replication crisis, especially. Of course, this is still done by the same lab, so we're also looking forward to other labs replicating our findings. But then if you look at the neural level, and this is pointed out here for self-pain, so when people are in pain themselves, you get also this reversal of the amplitude. So the effect disappears on the level of the ERPs. So that's an extension of the behavioral experiment and a specific relation to the P2 and the neural effects. So to sum it up, in the first part, we have 4 interwoven studies with independent samples, different methods showing converging evidence that experimentally reducing first-hand pain also reduces self-reported empathy for pain. That these effects are associated with reduced signatures of neural processes, both using ERPs and fMRI. And on the neurochemical level, I think that is the most exciting finding. Still, by using an opioid antagonist, we can block the effects both for first-hand pain, so pain experienced in the self, and empathy for pain. And that suggests a rather direct causal role for opioidergic mediation of pain. So to sum up or to conclude for the first part, for the affect sharing component, there seems to be a functional correspondence of the neural and neurochemical processes involved in first-hand pain and empathy for pain. That would confirm models that empathy implies an experience of others' emotions in one's own affective pain systems. And this may have important implications for prosocial behavior, but also for biasing it. Because if you're given, for example, or if you're taking painkillers, at least that's the assumption, you might also have an effect not only on your own pain, but also on sharing the pain of others. Now, some of you might have heard about this research. After we have published our findings, there was another study in a high-impact journal in eLife by Thor Weger and his group. And they basically tried to show that there is no such thing as shared representations between self-pain and other pain. However, there is something that could have been criticized, as in any study, of course, which could have been the paradigm they have used, because they did not have electrical pain stimulation for self and other, but they asked participants to take such pictures as this one here and imagine to be in pain themselves, so that they imagine to feel this pain inflicted by this hammer, for example, hammering on your thumb. So that was a slight limitation in the experimental paradigm. More recently, in the same high-impact journal, there was another paper with a much larger sample size, and with Thor Weger also being a co-author, that kind of reversed this picture a little bit. So in this paper, they have identified shared representations in a specific subcomponent of the insula. So predominantly the middle part of the mid-anterior part of the insula. So there is, let's say, a debate, or less of a debate now anymore, that there could be something like shared representations also if you use other analysis approaches. Remember that we only used region of interest analysis approaches so far, so that could have been criticized. And there's also converging evidence from the animal literature. So this is a paper by Christian Kaiser's group, where they basically used a very similar approach than we did. First, a neural correlate measure with electrophysiology in rats, and then psychopharmacological intervention with muscle microinjections into the ACC. And they basically find also shared representations on the electrophysiological level and the blocking of the effects with the psychopharmacology. So the take-home message of the first part is that there is converging evidence that something is shared. What exactly that is and how relevant it is for behavior is still a question to be explored, and maybe some of you get excited by this research and follow that up. Now let's get to the second part, and I have only 8 minutes left, so let's see how quickly I can zoom through this one. Empathy and self-order distinction. So what we talked about so far is basically how can I share the emotion of the other person. But in many cases, we don't really share what others are feeling. So some humoristic elements, some elements where you cannot really share what the other person is really feeling, or this picture here. That's usually quite upsetting. In a real audience, I would now see some puffs and hisses like, that doesn't look so good. But if you look at the actual expression of this person on the picture here, this person is not in pain. You don't have to be an expert in pain readings. For whatever reason, there is no pain in this person. It just looks painful to you. So you have to regulate between what is the person in front of me actually feeling and what would be my own experience would I be doing something like this. And that's what we wanted to look at. And for that reason, we developed an experimental paradigm that allowed us to disentangle congruent and incongruent emotional states in this case. Very briefly, how did this paradigm look like? We had, again, 2 participants coming into the imaging environment or behavioral study. And one participant would undergo, for example, an aversive stimulation of their hand and see a picture on the screen. So imagine you're the participant, you're lying in the scanner, you're seeing a picture of this stink bug, whatever the biological exact name would be. And you're touched by something that feels as if this is crawling over your left hand. The second person, the second participant, would see something like this snail or slug and would be touched by something that feels like being touched by a slug or a slug crawling over their hand. This would be a congruent case. Congruent between you and the other, and you would have to judge how does it feel for the other participant to be in this situation. The interesting part is the incongruent one. Now you are touched by something that feels pleasant, like the touch of a rose going over your hand, and you see the picture of the rose, and the other participant is still being touched by the slug and feels something like a slug crawling over their left hand. And this is the interesting part. So the egocentricity bias would be the difference between the incongruent and the congruent condition, the trials between incongruent and congruent conditions. So keep in mind that what the other person feels is always the same. The only thing that differs is in which affective emotional state you are in. So the idea was here to investigate how can we overcome the interference, the emotional egocentricity bias from being in an incongruent state to the other person. We also did that in the pleasant domain. Let's jump right into the results here as well. We had a series of experiments as well. I think that's always recommended, or that's what we always do in our lab, to have converging evidence, multimodal from different experiments, different samples, different approaches. On the behavioral level, we observed a small emotional egocentricity bias. These are the patterns you can see here. Basically, to summarize this very briefly to you, what this tells us that participants are generally very good in overcoming emotional egocentricity, in overcoming this interference between their own feeling state and the feeling state of the other person. But there's a slight kind of remainder of emotional egocentricity. So there is interference, but in statistical terms, it's low to medium effect size. When we decrease the response time, so when we in a way stress people to deliver their responses, this emotional egocentricity bias increases. So that's interesting from a cognitive and regulation perspective. Now, what's important for the talk here is how is that related to neurocorrelates or maybe even mechanisms. So when we use this paradigm to pinpoint the neurocorrelates, we got in 2 experiments with an internal replication. Part of the brain that is called the right temporal parietal junction, the more anterior part, which is the right supramarginal gyrus, is active consistently when people have to overcome this emotional egocentricity. The problem of this approach, again, is, however, that fMRI is only a neurocorrelate measure. So it could be either that this area is really helping us to overcome the emotional egocentricity, or it is actually instantiating the emotional egocentricity bias. So because I have a bias, this area is active. How can we solve this problem? We used TMS, transcranial magnetic stimulation of that area in an independent sample. And we used an inhibitory protocol. Inhibitory means that in a way you're increasing the noise level in the area you're stimulating, and that should have effects on the behavior, negative or positive ones. That's what we want to find out with this study. What we did find out was that we have an increase, almost 3-time increase in emotional egocentricity when we inhibit our TMS. And that tells us that SNG is involved in overcoming emotional egocentricity, because if it would be the other way around, it would instantiate the bias. So that is also, I think, quite a nice way to combine methods to get a close look at mechanistic insight. That, however, maybe will not be so exciting for the cognitive psychologists or the cognitive scientists amongst you, because we don't just want to understand where something happens in the brain, but also what and how. And to get at that question, we tried to compare the area that we found activated to other areas also associated with self-order distinction. That was unfortunately not very conclusive. I mean, we identified a genuinely different subdivision or function in the RSMG, but that doesn't tell us yet what exactly is happening. What did tell us something about the how is a connectivity analysis. So this analysis does not look at where in the brain something is happening, but in a way to which other areas of the brain, a brain area is talking to more in a certain situation. And what we found here, it's a bit of a complex slide, but I will walk you through. So this is the area that we identified with fMRI and also stimulated with TMS. And what you see here is the increased connectivity with this area when people are overcoming emotional egocentricity. On top of that, we have plotted some sort of localizer study of the contralateral to the left hand S1, somatosensory cortex 1, or primary somatosensory cortex, and secondary somatosensory cortex. And you can see an increase in connectivity with these areas, contralateral to the stimulated hand, and with the visual cortex. What you don't see here is that this is bilateral. So one way to look at this is that overcoming this emotional egocentricity or self-order distinction in the affective domain is achieved by some sort of increased interaction between private and public sensory channels. Private means everything that only I can directly perceive. So tactile input, for example, interoception. Public is the visual domain. So in a way, you could think of overcoming emotional egocentricity as a multisensory social integration. And this is something we're currently following up. Since time is running out, I will not talk about the aging-related aspects. I would just want to come to the conclusion. Sorry for taking a bit longer than usual. It's always a bit difficult without any input, any direct feedback. Anyway, so this is the summary. So of the part 2, empathy not only requires a mechanism for sharing each other's emotions, but also for keeping them separate. So that's a self-order distinction. Right SMG seems to play a causal role in this respect, and one possible mechanism is a multisensory integration of self versus other related information. And the age-related changes is something I will have to keep for another time. General wrap-up: empathy is a complex, multifaceted, and dynamic phenomenon. So looking back 15 years of research in the domain of social neuroscience, I think we have learned quite a lot. That is not just an automatic, nor only a controlled phenomenon. And take-home message part 2 in that sense is that affect sharing and self-order distinction are two important components that generate nuanced responses in how we share the affect, the emotion of others, how we can empathize. And understanding the mechanisms, I think that is the main message of the whole research program, will also hopefully help us to understand complex related phenomena such as pro-social moral behavior, how we react to others under stress, psychopathy, clinical conditions. So this would be another talk. That's basically the graphical summary, and these are the people I would like to thank and also the funding agencies, and thank you for listening, and I look forward to a stimulating discussion. Thank you.
Thanks, Klaus, a lot for this really fascinating insight into empathy research. And I also read questions here, or I try to give up the questions to you. What exactly does the subject see in terms of the other pain is one question.
Yeah. So what they see is, depends a little bit on the exact setup, but usually we try to keep it quite abstract. They don't have direct access to the emotional expressions, but they are given information that the other person is now in pain or not by abstract symbolic cues. For example, when they are in pain themselves, they see a red cue flashing on the screen and they get at the same time the painful shock. When it relates to the other person, an arrow points to the other person and there's another red cue that is flashing. So the participant from which we are recording the brain activity knows that the other person is now in pain. And the reason we do that is a bit technical. So we try to disentangle empathy from emotion contagion. So basically just seeing the emotion expressions of the other person creates already something in the observer. And that is a less pure measure of empathy, at least we think so.
Thanks,
I
Klaus.
hope that answered the question.
Yeah. I would suggest, since numbers of questions are increasing, we make the answers quite short. And if somebody would like to speak in person, please just turn your video on and let me know that you have a question. We will see that then. The other question of this person was, does it matter who the person is to whom you empathize?
Yes, so there are some studies that show so basically the main assumption is the higher the perceived similarity or the higher the connection, the stronger the empathic response. However, that is only replicated here and there. So it's not so clear. It makes intuitive sense, but the real evidence for that, I would say, is not missing, but it's controversially discussed. So in principle, yes, but in reality, maybe not.
Okay, thanks. How does this translate in the case of intraspecies empathy and perception of pain is another question of Ayushi.
Yeah, if I understand correctly, the question is, does this matter only for empathy from humans to humans, or maybe also empathy from humans to animals? I would say not fully, but partially the mechanism is probably the same. But then you have this similarity issue. So let's say you empathize with let's say a cockroach. So something that is not very similar to you, might not even have pain. You could expect some modulation of that compared to empathy for other mammals, especially if they're maybe cute or we can relate to them. So this is something that is quite underexplored. There are very few studies that, for example, try to compare empathy in vegans, vegetarians, and omnivores. And also here, I would say the final word that this is not really so clear. It's quite a complex question.
Okay. Another question, do you know if the functional correspondence observed in between self-pain and empathic pain, where's the question, could be extended to the feeling of compassion? Is compassion different on the neural level than empathy, as many social scientists such as Paul Bloom suggest?
Yeah, that's unfortunately a question that would require some deeper reasoning, let's say. But the short answer is yes, empathy and compassion are two different things. So empathy is sharing what the other person feels. Compassion is also caring for what the other person feels. So you can construct, or you can think of situations where you have a lot of empathy but no compassion, and also the other way around.
Yes. Paolo Sola, in light of your results, how should a depressed subjects behave both from a placebo, naltrexone, and self-other perspective?
So that's a very interesting question. So we're also performing research on that, not with placebo yet, but basically there's a general blunting of affect in depression, and the prediction would be that they should also show less empathy for the pain of the other person. And we have some indirect evidence that this is actually happening.
Yes. The next question, we know from autism spectrum disorder research that sensory perception quotient test yields a quasi-normal distribution in neurotypical people. Was that controlled for in the visual tactile experimental design?
That specifically was not controlled for, no. So I can follow that up, but keep in mind that here those were all neurotypical participants. So I don't recall from the top of my head whether we also had an autism trait questionnaire. I think so, but I would say this should not affect the results.
Okay, and the final question that I can see here is, why would there be hyperalgesia on getting psychopharmacological intervention? Shouldn't the two groups be comparable after the intervention?
So that maybe was a bit imprecise for me, hyperalgesia compared to the other group. So just keep in mind that both of them are given the placebo analgesia induction. So in a way, they think they are giving a painkiller. And if they're completely level, it would mean that there is no effect of the psychopharmacological manipulation. However, one group reported more pain than the other. Now, if you compare this group that reports more pain to the control group, they are level. So in the end, they have hyperalgesia compared to the matched group with placebo analgesia induction, but not with the opioid antagonist. But they're on the same level as a control group that has not undergone any placebo analgesia or naltrexone manipulation. Hope that answered the question.
Okay, here are more questions. Wait a minute, we still have a few minutes. I just lost my screen.
I can read it. How well do reported reduction of attribute? Yeah, I can drive. So how well do reported reduction of attributed pain in others correlate with the electrophysiological findings? Would subjects with low reported attributed pain in others still show similar electrophysiological effects as subjects with high reported pain in others? Excellent question. We don't regularly find these correlations. That might be to do with the sample size, but also the question whether you find a linear relationship between pain reporting and pain activation. But I guess the question points at kind of looking at different subgroups of high and low pain sensitivity. And there is some evidence that this plays, of course, a role, whether you're pain sensitive or not. Kalina Michalska, hi Kalina, great talk, how important, thank you, how important it's jumping, how do you think specificities for empathic pain? That is, if I decreased pain in one bodily area, but observed others' pain in a different area, would that show less overlap, or is it about general level of bodily pain? Excellent question. I can answer that with our recent neuroimage paper, Hartmann et al., where we used placebo analgesia for the somatosensory component. So there we find no evidence for manipulation or modulation. So indirectly, that answers your question in the sense that it seems to be the general affective motivational component, irrespective of which body part has created that. Our original hypothesis was different and was pre-registered, but the data point in the other direction, so that it is not specific to the body part. Have you found structural cortical variations associated with empathic capacity? There is a paper by, what's his name again? So the last author is Tanya Singer, Boris Bernhardt. He's now at MNI, I think, Montreal Neurological Institute. And yes, there are structural cortical variations. There's also a couple of morphometric studies, and some of them are in the areas that we talked about today, so the anterior insula and the MCC. Jumping around.
There's another one. Do you think cognitive experience could invert the connection between tactile visual pain perception, e.g. having their blood drawn? Some will feel less pain when looking at the spot, while others feel better to look away. What might cause such ambivalent experience?
Here I have to say I lack a bit the context, what your real question is. I don't know, Ute, whether you can help me, but I mean, basically ambivalent experiences across participants, individual differences can have many different reasons, but let me read again. Do you think cognitive experience? So if you have experience...
I think it's focusing versus drawing your attention away from it.
Mm-hmm. Well, I mean, attention plays, of course, a big role in the whole process. So the more attention and the more memory and the more knowledge you have about a certain situation, the stronger the modulation of the empathic response. So I'm not sure that is really a good answer or a satisfying answer, but that's as much as I can say at the moment.
I think we made it perfectly in time. Is there, I can see 2 more minutes, but is there any urgent question? Please show up in the video or ask your final question. If not, then I would like to thank our speaker again for this really very nice talk. Here's one last. I don't know if we have enough time, but
yeah,
modulate.
I can read it. One of the recent studies suggested transient emotional episodes modulate the neurocircuits of pain and empathy. Have you observed any correlation between your results and the participants' pre-experimental positive and negative dispositions if you have taken them into account? So basically, you're asking whether trait individual differences in empathy, self-report correlate with these findings. Also here, let's put it this way. So we and others find such relations on and off, but it's not as clear-cut as one would expect, and that might again have many reasons. And one of the reasons is, of course, that the self-report questionnaires measure something different than the state responses that we see in the scanner. So some people, let's say, might want to present themselves as very empathic, while in reality they're not, and the other way around. So that just creates noise. And then with the usually small sample sizes, although we had 100 subjects in the fMRI study, it's easy to see that sometimes you don't see them. Theoretically, of course, you would predict that. So that people that self-report higher trait empathy show a stronger modulation.
You don't have to apologize. We're happy that we were able to answer all the questions. Thank you again for your participation. And Klaus, thank you again for this nice talk, and enjoy the rest of the conference and participate in other keynotes as well as symposia. Thank you and bye. Have a nice day.
Thanks a lot, everyone, and safe and healthy. Bye-bye.
So because there's plenty of time, I will not interrupt you. So if you take 6 or 7, that's fine.
Okay, sure.
If you're taking way longer, then I'll interrupt you. Otherwise.
Yeah, no, we'll keep that in mind. Okay.
Hello, guys.
Hey.
Hello.
Hello.
So can you tell me how to pronounce your name? Do you want me to say?
Yeah, Arefe. Yeah.
Just Arefe.
Arefe Farahmandi. Farahmandi is my last name. Don't care about it. Just Arefe. Call me Arefe.
Well, I'll call you Arefe, but when I introduce you, I want to also use your last name.
Yeah. Arefe Farahmandi Najaf Abadi. I think there's no need to call Najaf Abadi. Just Arefe Farahmandi.
Okay. Arefe Farahmandi.
Yeah, exactly.
Okay.
Yeah.
And you are at University of Tehran?
Yeah.
Are you a graduate, a PhD student, postdoc?
No, I'm master student.
A master student.
I will graduate soon, yeah.
And you have a presentation that's about 5 minutes long, give or take?
Yeah.
Okay, yeah. So I'll just let you speak, and I'll only interrupt you if you go,
Okay, just tell me what time exactly I should start.
Yeah, I'll start with the introduction quickly, and then.
Okay. Yeah.
And then.
I think I'm first
I'll
speaker. Yeah.
So yeah. You're the first speaker, exactly.
Okay. Yeah.
So I think we're probably live this whole time, but yeah, let's just wait another minute until the official start time, and then I'll introduce everybody. And Marta, maybe you can turn off your video right now, and you can turn it on for your talk.
Okay, can I start or?
Yeah, give me a second. Okay, so it is time for us to start. So let me just introduce myself first. My name is Dobi Rahnet. I am the MC for this session. And we have 3 wonderful talks. They're all about the function and organization of the temporal lobe in monkeys as well as in humans. It is a wonderful privilege to be the MC for this session. And I think it's a great example of how NeuroMatch is a truly global conference. Our 3 speakers here are from 3 completely different parts of the world. So we have somebody from Iran, somebody who is working in the Netherlands, and somebody who is working in India. And I myself am based in the US, as well as the back end here, who is going to be doing a lot of work behind the scenes, Gauri. She is in Arizona. So have a truly global conference here. So I just want to say these are going to be interactive talks. So the speakers are going to talk for about 5 minutes or so, and then we'll have time for questions afterwards. Do ask questions in the Q&A. We prefer that over questions in the chat, because in the Q&A you can also upvote them, and we'll ask the question from the Q&A first. There's no stupid questions, so please, this is supposed to be especially interactive, so please ask and participate. All right, so without further ado, let's go to our first speaker, Arafe Farahmandi from University of Tehran, and she is a master student who is going to tell us about modeling the temporal dynamics of neurons in the IT cortex.
Yeah, thanks. I'm Arafe, and I wanted to speak about temporal dynamic of neurons in the IT cortex. Let me start. And yeah, I think that's okay. Yeah, that's my screen. You can see all. Okay. Let's go. I want to talk about modeling the temporal dynamic of neurons in the IT cortex. Our project is based on experiment which is done on 2 monkeys. We have a rapid serial visual presentation in our task, and as you can see, we showed monkeys for 100 millisecond blank gray screen. After that, we show our image, which I will explore more fully, and it takes about 300 milliseconds. After that, again, we have a blank gray screen. At the same time, we have electrophysiological recording from IT cortex of those monkeys. And, we have different category of image. They are in different abstraction level, because we just want to say wanna see that whether they are encoding in multiple time course of our neural response or not. And as you see, images are divided into different category: animate, inanimate, body, face, and vice versa, which I will explore more in the next slide. Our goal is to design a model which gets our image and produce the neural response, artificial neural response for us. So we do this work into 2 steps. First of all, we use pre-trained ResNet. It is combination of convolution layer to extract the feature vector of those image for us. And in the second phase, we use partial least square regression, which use those feature vector of our image and trained by our real neural response. And after that, it can produce our artificial neural response. As we can see, the results are surprisingly great, because we can see here, for example, we have 2 sample image, and here we have the temporal dynamic neural response of our real neurons, which we record from IT cortex of monkeys. And here we have artificial neural response. As you can see, they are really similar to each other, and that's a really great for us. In the next step, we calculate the correlation between neural response and artificial neural response in different categories to see what is the value of correlation. And as you see, we have approximately about 80% correlation in different categories, and this is a really great result for us. In the other step, IT cortex is responsible for categorization. So we expect that our real neural response is able to classify different categories with each other. So we do some bootstrapping and classification on our real neural response to see what is happening. And as you can see, the blue one, the blue plot is using those real neural response, and it is a normalized accuracy of our classification. 0 shows exact time that we show image for the first time after blank gray screen to the monkeys. And as you see, this is increasing here. It's exactly the time course which our visual information of image is received on our RT cortex. So the accuracy is increasing, and the orange one is our artificial neural response. I mean, it is the accuracy of classification using our artificial neural response. And as you see, its behavior is such a good, because it's also is have this trend, and we do this for different classification. For example, here we have body face classification, here we have animate inanimate classification, and also we can see here that it is our neural response which is able to classify different category, and also we have a little good accuracy for our predict and artificial neural response. In the next step, we have basic level advantage, that it is expected to be observed in our real data. It is our real neural response, and here is artificial neural response. As you can see, in our brain, recent electrophysiological studies have suggested that earlier and later neural phase of neural response can encode multiple hierarchical abstraction of our image and our visual information. So body face is our mid-level abstraction of our visual information, and animate inanimate is superordinate level of abstraction. So we just anticipate that it should be observed that in our artificial neural response, the earlier phase of our neural response is responsible to classify the mid-level of our image. And as you see, this is really great, because we have this observation here, and you can see the peak time of our accuracy in body face is sooner than peak time of superordinate level of abstraction, and it is really similar to our real neural response. So that's really great for us. It's finished, and I'm just here to answer your question.
Thank you. That was wonderful. So if you have questions, please put them in the Q&A session, in the Q&A of Zoom. So we have a first question right now. How do you find the peaks? So Arafa, can you? And maybe,
yeah, exactly.
Maybe you can keep screen sharing and maybe leave it to one slide that is informative for people who are a little late.
We can. Okay, about peak. That's a good question, because we just have a lot of methods for calculating the peak, but here I use 90% of our response. The first time which we arrive at 90% of our peak value of plots, we name this time as peak time course of our data. There are different definitions for that, but we use here 90% of peak, I mean the max value of our accuracy. And after that, we just calculate the first time which we can arrive at this point.
All right, thank you. So then the highest upvoted question so far is by Michelle Green. Not clear about how the peaks were, oh, it just went a little, so not clear how the peaks were found for the basic level advantage for real neurons. They actually look a bit early. Can
I see the question?
Yeah, so if you click on the
I
Q&A.
should stop share screen to
Sure,
see
yeah,
the question. Yeah, not clear about how the peaks were found for the basic level advantage for real neuron. They actually look a bit early. I don't know if it's early, but as we investigate about that, we really found that, you know, it's about I think about 100 milliseconds after showing the image. So I think that's good. And I don't know if Michelle can explain about it. I think that's really expectable, but I don't know what is the normal range that you mean.
And Michelle, I just allowed you to
talk.
talk.
Hi. Yeah, what I meant is very similar to the previous question. When you looked at the peaks for the left-hand graph that you were sharing, it looked like the functions of the decoding accuracy kept increasing over time. And so it seemed like the peaks would actually be later on, and that the pattern would actually flip, that the animate-inanimate distinction looked like it was a little bit earlier than the face-body distinction for the real neurons, but not for the artificial neurons.
You mean the difference between, let me share my screen. You mean this, yeah?
Yes.
Yeah, that's really that's really strange for me too, because I think it should be like here, yeah, but I don't know. That's our result, and I should work on more. But we have a lot of methods here. I just use 90% of our accuracy, but maybe I can calculate it in the other methods to achieve a better value for my peak. But that was our observation, and yeah, I agree with you, because I just see that, and it was surprising for me, because I really anticipate that it is in this time course.
All right, great. So there was a question that got marked as answered, but actually wasn't addressed. So you have negative accuracy on your axis. I think the issue is that you're showing normalized accuracy. So maybe you can explain a little bit how you normalize the accuracy.
Sure. We calculate normalized accuracy, and it is based on our the accuracy that we have in our baseline, and baseline is defined the time course, the 100 milliseconds after showing image. This accuracy is our baseline accuracy, and we just normalize our accuracy based on this value. I mean the mean value of accuracy in our baseline time.
All right, wonderful. And one last question that we have, and I think we'll be out of time afterwards, by Zishan Kadir. You mentioned around 0.8 correlation among different categories. how this is advantageous for your classification problem?
It's not beneficial for our classification. It's the first phase of our results, because the goal of our project is to design a neural network that is able to predict and produce the neural response. And after that, we just calculate correlation of artificial neural response with our real neural response to see whether they are correlated with each other or not. And they don't I mean, that's not related to our classification problem. We just use classification to see the categorization of our neural response, because IT cortex is responsible for this task. So we just want to test about our artificial neural response to see if they are true for our artificial neural response either.
All right, so great. There's actually one more question from the chat that I would like to ask you, and I was actually wondering about that myself. So this is from Eugia Penk. Could you elaborate on how the artificial neural responses were calculated over the time domain? So for the artificial responses, how did you get?
Yeah. You know, as I said, we have two steps. At the first, we just extract the feature vector of those images that we showed to the monkeys. And after that, we use partially least square regression. It gets our feature vector that is a vector, and after that, give us the output of, I mean, the time series of our response. And, you know, a regression is able to do this, but in the next step in our project, we just want to inject some dynamics in our ResNet, because in this work, we don't have dynamics in our ResNet, but in the next step, I just wanted to inject some dynamics. I mean, feature vectors are changing in time course, because the visual information that's received in the IT cortex of monkeys is changing over the time. So we just wanted to do this work for future.
Wonderful. Thank you, Rafa. That was great.
Oh, thanks.
So,
thank you. Yeah.
So next up, we have Marta Pollo, who is a PhD student in Maastricht University, and she's going to talk about computation-based feature representation of body expressions in the human brain. Marta, take it away.
Yeah. Can you see my screen?
Yes. Yes.
Okay, perfect. Thank you very much for the introduction. Indeed, I'm coming from Maastricht University, from the Brain and Emotion Lab, led by Professor Beatrice de Gelder, where we mostly investigate body expression perception. As you may know, body expression is at the conflux of questions related to object category perception, so a body as an object category, also action representation, and affective or emotion coding. In our lab, what we try to convey is, what we try to do is integrate these different lines of research, all related to bodies, to further investigate, to further understand body expression perception. In particular, what we do is to go beyond these high-level constructs, such as emotion category or action category, to further understand emotion perception in the bodies. In particular, this brings us to the current study, where we look into the lower level properties of the body to see, for example, if postural and kinematic features already convey emotions. And as well, if these features are represented in the brain. To pursue these goals, we use whole body movement videos, as you can see in the picture, representing an angry, a happy, a neutral, or a fearful body expression. And before computing these features, we had to estimate the position of the body main joints, for which we use a post-estimation library called OpenPose that gave us the X and Y coordinates of these 18 key points that you can see on the screen. and did so for the total duration of the video, which was in our case 1 second, that consisted of 25 frames. Once we had this information, we computed several kinematic and postural body features that have shown to be relevant in previous literature from studies doing descriptions of body expressions and also behavioral studies. But none of them were computational at this one. Within the kinematic body features, we computed velocity, acceleration, and vertical movement. Within postural body features, we computed symmetry, surface, and different measures of body contractions, such as the angle between the body segments or the distance of the hands and the feet to the head, a feature that we call limb contraction. Once we had these features calculated, we wanted to see whether we could see differences at this feature level between emotions. And for that, we computed representational similarity analysis. Here are the results of this analysis with one matrix per feature. This matrix basically represents pairwise comparisons of these feature values between videos. Here you can see in the diagonal that are comparisons between similar videos, and off-diagonal values are comparisons between different videos, with blue indicating similarity and yellow dissimilarity. And these matrices are arranged as in this emotional category matrix, with anger here, followed with happiness, neutral, and fear. And what we can take from this analysis is that postural rather than kinematic features represent better the emotional structure. For example, here in symmetry, it's really clear that neutral body postures are quite different from the rest of the body expressions. The same can be seen here in the matrix of limb angles, along with a difference between the fearful body expressions and the rest, which was also present in the feature limb contraction. This is an overview of the whole project. So far, I just talked about the post-estimation, feature calculation, and analysis, but we wanted to see where these features are encoded in the brain. And for that, we performed an fMRI experiment where we show the same videos to participants where they were lying in a 3 Tesla scanner. And subsequently, we correlated these neural RDMs to the feature RDMs to see where these features are encoded in the brain. This is a specific case, a specific example with the feature limb contraction that I said before, it codes for fear. And we saw that this feature is represented in several areas spanning action observation, motor preparation, and affective coding networks, including the amygdala. All have shown to be also related, all have shown to be related to fear. And to conclude, what we, in our study, we went beyond the study of emotion at the categorical level and showed that emotion is already conveyed at the mid-level feature level. If you want to know more, you can find this study already published in Cerebral Cortex, and we also have another study relating the same features to subjective ratings on postural and kinematic information. Thank you.
Thank you, Marta. That was wonderful. All right, so if you have questions, please type them in the Q&A. I know this is very short talk, so it's hard to type at the same time. In the meantime, let me ask a quick question myself. So you, did you try to, at any point, classify these emotions, these 4 emotions from either the videos themselves, from the different features that you extracted, or from the brain responses themselves?
Yeah, we perform several kinds of analysis. We also perform one type of analysis where we just input to an algorithm a classification regression tree all the key points. And we saw, for example, that actually the hands are the ones driving the perception of emotion. For example, we perform this kind of analysis, and we also perform another analysis using this matrix of coding for emotion, and so that actually emotion is distributed across the brain as well. Yeah, we did several kinds of analysis apart from the ones that I'm showing here. Yeah.
All right, that's great. So while waiting for other, there's a question by Jiwan Yon. Sorry, the slide went too fast. What is the lowest level in slide 8?
Yeah, the lowest level are features like, for example, luminance or like motion and pixel intensity and so on. Then we have the mid-level features where we already have like postural and kinematic information, but we have to understand that there is actually not a framework out there of analysis of body expressions. So it's actually this study was a bit like swimming in the darkness and trying to investigate these mid-level body features and how they convey emotion.
All right, thank you. 2 more questions from Carly Sombrick. Do you think the association between emotion and posture is learned?
That's a good question. It could be, I guess it could be learned, of course, yeah. Like in different ethnicities, for example, probably have different ways of expressing emotion, and that would lead also to different mid-level body features, of course, yeah.
All right, great. 2 more questions. First, Nayun O asked, how were the emotions labeled in the motion videos?
So we had basically 4 categories, and we decided to have angry, happy, neutral, and fear. And we basically instructed our actors, volunteers in reality, to perform this kind of emotions. So we predefined these labels of the videos.
Okay. Another question from Marco Gandolfo. Thanks for your talk. Do you think there's something special about fear that other emotions don't have? And if so, wouldn't that speak to some high representation of emotion other than mid-level features?
That's a really good question. I think the brain is wired for survival, for adaptive behavior, and indeed fear is a very valuable emotion for that. And if you think about it, the approach of mid-level features for fear perception makes a lot of sense, because sometimes we don't see perfectly a situation, but we still know that we should feel fear. For example, I'm walking in the darkness in the woods, and I see something that could be like a bear or something, and even though I'm not sure what I'm seeing, I'm already perceiving fear. And probably the mid-feature level, where we don't have to really interpret the categorical level, already informs us that we should prepare for an adaptive action.
All right, thanks. A clarifying question about the similarity matrices. How did you compute them? Did you use Pearson correlation? This is from Pablo Martinez Cañada.
For the feature matrices, I use Euclidean distance, and to correlate the feature matrices to the searchlight resulting matrices, I use Spearman rank correlation.
Maybe I can just do a follow-up myself. Euclidean distance between what and what exactly?
So we had all the feature values within one video, and we performed the Euclidean distance between pairs of videos.
Okay, wonderful. All right, two more quick, well, we'll see if we have time for both of them. So Yujia Peng asks, in the middle level, do you think low level motion cues or global body posture drive emotion categories more?
Sorry, can you repeat again?
Yeah, so in the middle level, do you think it's low level motion cues or global body posture that drives the emotion categories?
Well, here what we found is that actually postural information may be the one driving the emotion perception per se, rather than motion. But I cannot speak for larger data sets or different data sets.
Okay, and I think we have time for one final question. This is from Isaac Moran. How do you distinguish the neural responses associated to emotion from motion processes? So just how do you know it's movement? How do you know it's emotion rather than movement?
That's a really good question as well. So basically, we are looking at basically we have in our stimuli neutral and emotional body expressions. And if we see that a feature is differentiating, for example, between neutral and the rest of the body expressions, because both of them are moving, we could basically conclude that this feature is differentiating between emotions, because both of them, all our stimuli are dynamic.
All right, great. Well, thank you so much again. Here's my applause right here. And next up, we have Avisha from the Indian Institute of Technology in Kanpur. And Avisha is actually a fourth-year undergraduate student. So thank you so much for being brave enough to present to a global audience. So she's going to talk to us about shared neuroactivity patterns between facial expression and shape recognition. Avisha, take it away.
Hey, so actually it's a group project, and like my other friends will start first. I'll just share my screen. Yeah. Abhinav, you can take over.
Hello everyone, I am Avnav Singh and I welcome you all to our poster Shared Neural Activity Pattern Between Facial and Shape Recognition. Along with me, my group mate Avisha and Hardik will also present you and you will meet them later in this talk. We came together from different places of the India in Neuromatch Academy, which held this summer. And this project which we are going to present was a part of our Neuromatch Academy project, which we did in limited time and with limited resources. So this project would not have been possible without the help of our mentor, Dr. Nidhi Sitapati, and for her support, without her support, and she was our NMA mentor actually. And our pod TAs, Elish Dipani and Anandita Bhattacharya. So Avisha, can you change the slide? So after brainstorming during NMA, we came to a scientific question that can we differentiate between facial expression recognition and shape recognition by just looking at the fMRI activity from the brain, following which we hypothesized that facial recognition mechanism shares some shape recognition steps in visual cortex area. Abhishek, please change the slide. To test our hypothesis, we used emotion tasks fMRI data of the human connectome project, which were given to us during NMA project. The task was to match either the facial expression or the shape from the two options in the same category. As you can see in the figure, the participants were given one picture and they have to match either of the two pictures from the bottom of the screen. The participants were given 3 seconds cue before each of the tasks, in which they were told that what they are going to see, either face or the shape. There were 339 participants from which the data were collected, and there were 2 runs for each participant, and each run contains shape task and face task. So this was the overall structure of the data set which we get during NMA project. And now what we did with the data set will be explained by the Hardik. So Ardi, can you take on, please?
Yeah, thank you. So to start off with the data visualization, we use tsne as a way to reduce our dimensions. So initially we had 360 features, so that is quite a large feature space which we need to somehow visualize to get some sort of intuition on the data sets, on the data points. So we tried tsne, and it stands for like the stochastic neighbor embedding. And what we observed was that if you look at the on the legend of this of the figure, we can see that the tasks corresponding to the face shape and the blank frames are quite nicely separated, and also that we are embedding that into a 2D space. So the tasks are separated, and also that that the number denoting them is the timestamp of that specific period. So that means that there is some sort of differentiable component which we can further analyze, and to finally explore that, we moved on to functional connectivity. And for functional connectivity, basically it's like the pattern of anatomical links of the statistical dependencies of each of the parcels. And so functional connectivity, we basically measured the Pearson's coefficient for a parcel corresponding to each of the different parcels, and like for their generated bold signals. And what we observed is that in the facial data task, when the person was given to analyze the facial data, we could see that in the lower posterior view, in the lower region, there's a lot of activity. And similarly for the shape, correspondingly for the shape task, we could see that in the posterior view there was a lot of interconnected activity. But also what we observed was that there is a reason that these are not very intuitive at first glance, because what we thought was the face task should contain lesser than the shape task, because what we think that shapes are more rudimentary compared to faces. But the reason for that is that because of the way this graph was computed at a specific instance of time, the underlying density is on the face task is actually more than the shape task. However, they don't look the same this way. Moving on to how we actually modeled the data, we used a very popular regime called the multivariate pattern analysis method, where basically the traditional brain mapping usually treat brain as the outcome and the task as the predictor, and there isn't really any accounting for the interaction in between the voxels. So basically MVPA tries to account for this interaction interdependent between the voxels, and it usually consists of three major steps. So first is the feature selection. Which features do we actually need from this huge 360 voxel array? For that, we used ANOVA, which is a feature selector which is commonly used in scikit-learn. And then from selecting those 5%, we basically chose the top 5% features out of the 360. So in the end, we chose 18 parcels out of the 360 parcels. And on these 18 features, we performed binary classification, for which we used the SVM linear kernel. And for that, we didn't do multi-class classification. We actually chose binary classification because that was more interpretable to us. And we did the task shape versus the none, and the face shape versus none, so that we have some sort of control and baseline to compare both of them to each other. Regarding the accuracy, we got around 82 and 80% accuracy for the face and the shape face and the shape model, but mainly the interpretation is regarding the accuracy is not really a measure which we usually take for classifiers, because it does not actually express the granularity of things like precision and if there are any true positive and true negative that are not that are underlying in the classifier. So if we look into the confusion matrix, what we see is that in the blank versus blank, our model performed good because there isn't really anything to actually determine from that data. But in the places which our model did not perform that great was when the true label is actually blank, but what we predict it's a face. So that is one thing where the accuracy plummeted. And also similarly for the corresponding for the shape task, if the true label is blank and the actual label is shape, that had a very low accuracy. However, corresponding to the other places, when it's a face and we predict it's a blank, similarly for shape and blank, those were relatively higher. So then we moved on to like our results, and that will be taken over by Avisha.
Okay, thanks. So yeah, finally, from the top 5% features, that are 18 parcels out of 360, and we classify them. And then since we used SVM linear kernel, so the linear kernels are interpretable, and the weights which we get from that model are the activations of different parcels, are the features, the activations of the features. And using those activations, we drew this graph of ventral view of left hemisphere, right hemisphere for both the tasks. And here we can see that there are mainly 3 regions which highly correlate even in shape task, FFC, VMV3 and PET, and here in the facial expression recognition task also. So there was a high correlation in few regions which was observed in both kinds of tasks. And hence the major takeaway from this whole exercise was that we could see posterior inferior temporal PET region, fusiform face complex FFC, and VMV3 in ventromedial visual area, which highly correlated with shape recognition task as well as facial recognition task. So we now know that both of these brain regions are very active during both kinds of recognition. And there were few visual cortical areas like V1, V2, V3, which were also highly active during both the tasks and were visible even in the top 18 features, which we selected using feature selection. So the conclusion was that there are some brain regions, which are common in both kinds of processing in our brain, and hence their neural activity pattern, they share a neural activity pattern. And since this is an ongoing work, we are open to suggestions, what we can do next, how we can improve our model, how we can improve the interpretability of our model. We have not taken yet the temporal aspect of the time series into account. We did not see that temporally which task happened first. So what was the corresponding activity in the next trial during that task? We did not consider all of that. And we did not even take account the behavioral data, what were the actual responses of the participants, and what was the reaction time in which they finally marked the correct response there. So these can be some further directions on which we can work on. Thank you. And we can take questions now.
All right. Thank you to all 3 of you. That was very interesting and very creative format. So here's my applause. All right, so I don't see question and answer so far. So if you have questions, type them in the Q&A box. In the meantime, that was a very interesting project, but I'm also very interested in just your experience forming this collaboration, from Neuromatch Academy. Can you tell us a little bit more about that? That's just very fascinating that you're able to form this group.
Yeah, so it was an interesting opportunity and first of its own in the pandemic. And we got it was a 3 weeks workshop and 5 days each week. And from the second week of the workshop, we started working on the project. So effectively, we worked on this project for 2 weeks. And we had to come up with our own hypothesis. What kind of questions do we really want to ask? Which kind of data sets are suitable to answer those kind of questions? And there was a lot of brainstorming. I met these guys, my group mates, in the workshop itself. So interacting with them, interacting with our pod mentors, and taking their advice and discussing, figuring out how to manage such a large data set, higher dimensional data sets. So yeah, it was an interesting journey, and we thought we should share it with other people.
Well, shout out to Neuromatch Academy. I think it's a wonderful thing that was done, and there's clearly results here. We have one comment, no question, just want to applaud them for such an interesting project. So, I think people really liked
Thank you.
Thank you.
project. Okay, one more from Lucy Lu. What does face expression specifically share with shape recognition instead of face recognition in the first step?
Sorry, I didn't get it. Let me just read
So
the question.
Yeah, you can see in the Q&A, and any of the three of you can take the question.
So what I'm getting is that what is the motivation of investigating these expressions? So our initial motivation to actually go into this route was that what we think for an intuition that faces are usually comprise of shapes, because if we break down face from their landmarks, and if we just take face and we see how we might actually process it, so what we thought was, if facial data is broken down into some sort of shape recognition, and then it is finally processed, because what we thought is that the whole the whole visual data is not just processed at the dimensions which it is incoming. It might be reduced to some lower dimensions, and that's what we thought, that faces could be broken down into shapes. And to prove that sort of hypothesis, we then looked into what is the activity in the brain for shapes itself. So to correlate this sort of relationship, we try to look into this data.
All right, thank you. So it is 12:46, so let's give one more round of applause by myself here. And another round of applause for all of the attendees and all of the presenters. Thank you all for making this so interactive and so interesting. And this is just the beginning of NeuroMatch. I think that was a great kickoff session for the interactive talks. Please attend the rest of the conference and enjoy.
In the chat window. Okay, so looks like we are live. So good morning, actually good afternoon, everyone, for people on the East Coast of United States. My name is Chen Xue. I am a systems neuroscientist from University of Pittsburgh. I study neuronal mechanism for natural decision-making. But as I read through the abstracts of today's talks, which heavily focus on synaptic plasticity, I am genuinely surprised at how, although we do our studies on very different levels, we are actually wondering about very similar questions. And I'm sure the audience will feel the same way as well during the talks. So please feel free to ask any question that interests you, regardless of your background. Even if you think it's kind of dumb, my job today is to be here and be the dumb guy for you. That's kind of the exclusive service I offer only during a pandemic. So, all right. With no further ado, our first speaker is Julian Hosbrock. He is going to talk about his computational work with Richard Nord at University of Ottawa, in which he will show us how a simple linear nonlinear model can explain a variety of synaptic dynamics. So Julian, please tell us more. I will stop my share so that you can come on stage.
Thank you so much, Cheng. I can't share my screen yet. It says it's disabled, the attendee screen sharing. And oh, no, there we go. Now it works, wonderful. Yeah, thank you, Cheng, and thank you, Ali. I'm really happy to be here today. So thanks for giving me the opportunity and for hosting this session. I'll dive right in. What I'm going to talk about today is short-term plasticity and how best to describe it in a computational model. So when I say short-term plasticity, what I mean is that neuronal communication typically is not static, in that a train of consecutive action potentials won't lead to the same postsynaptic response. And traditionally, people like to distinguish between between facilitating and depressing synapses, corresponding on whether consecutive spikes evoke increasingly or decreasingly large responses. But we were fundamentally motivated in this work by a few observations of experimental neuroscience that this dichotomy of facilitating and depression is not really that accurate. So just to give you an example here, this is a paper from Simon Chamberlain from 2014, where he showed that at a specific synapse in the hippocampus, if you lower the extracellular calcium concentration, you don't only change the initial EPSC amplitude, but you also drastically change the time course of facilitation from this more classical sublinear time course to a supralinear facilitation. And findings like this challenge this traditional dichotomous view of depressing versus facilitating synapses, and maybe suggest that there's something more to the story. So if you want to describe synaptic plasticity or short-term plasticity, the traditional approach would be something like the Zollix-Markram model, which is heavily used throughout the field. And in essence, what it tries to do is tries to describe the efficacy of a synaptic spike as the product of the available and the utilized resources at a synapse. So in other words, the available resources correspond to something like the available vesicle pool, and the utilized resources to the release probability. And then as we have a spike train, a presynaptic spike train, the available efficacy at each spike will go down and then recover again, and the release probability after each spike will go up. And then this product of the two, depending how you fit the parameters that go into these two differential equations, you can describe facilitating synapses and depressing synapses and a combination of the two. So here's the problem with this. This model is wonderfully elegant and simplistic in that it's a mechanistic model where we can take these parameters and directly pinpoint to a biophysical property they describe. But at synapses that are more complex, like the one I just showed you, we know that these phenotypes are the result of multiple interacting and independent biophysical mechanisms. Of course, we could start, adding factors to this model and adding a factor for each biophysical property that we want to describe. But as we add factors, we also sort of collect a catalog of parameters that we then have to fit to our limited experimental data. So in order to describe complex dynamics, these mechanistic models become very, very complex very quickly. So we take a different approach here, and we try to sacrifice some of this mechanistic interpretability and really focus on describing the phenotype. So in order to construct such a phenomenological model, the essential problem that we have is to get from a spike train here in the top left to a weighted spike train, which I will refer to as an efficacy train here in E. And E is essentially just a weighted spike train that then can be used to be convolved with some postsynaptic kernel, in this case a current, to give rise to a postsynaptic response. But how do we get from the spike train to an efficacy train? Essentially, what our model does is we take a presynaptic efficacy kernel and we convolve the presynaptic spike train with this efficacy kernel. And that will give us a accumulated efficacy train throughout the spike train. So this linear operation is then followed by a nonlinear readout where we take a sigmoidal readout of this accumulated efficacy. And we also add a baseline B. And then this nonlinear readout of the accumulated efficacy sampled at the spike times will give us an efficacy train. This might be a bit much at first, but essentially it's the following. We have a linear convolutional operation followed by a nonlinear readout of this accumulated efficacy. And what I want you to realize is that now, by changing the shape of this presynaptic efficacy kernel that essentially describes how the time course of the efficacy change in response to each spike. If in this case, it's just a mono-exponential decay with a positive amplitude. If we change the amplitude to be negative instead of a facilitating synapse, we will now get a depressing synapse. And all the way on the right, I took a kernel that is a sum of two exponentials with opposite sign and different time constants. And that will give us a combination of a synapse that's first facilitating and then depressing. Okay, but these are all, this is still this dichotomous view of the synapse. So what about more complex dynamics? So in this supralinear facilitation example that I showed you, we can model this by changing the baseline parameter. So because we have a nonlinear readout of the accumulated efficacy, there's a sigmoidal function, we can set here again, I take in E, top left, I take a presynaptic efficacy kernel, and then I convolve it with a presynaptic spike train. And then here in blue and red, I just set two different baseline parameters. And when we take a nonlinear readout of this accumulated efficacy with different baselines, we exactly get this distinction between the sublinear and supralinear facilitation. I want to take a step here and maybe show you another atypical example at the end if I have time. But there's one other thing I really want to focus on, which is that all these traditional models and this model, they treat the deterministic case of synaptic transmission, where we essentially consider the mean EPSC amplitude averaged across a lot of trials. But synapses are not deterministic, they're highly probabilistic. And we wanted to extend our model to this stochastic framework. So instead of considering the mean efficacy, we can also consider now a random variable, let's say y, and the postsynaptic response, here the current, is now determined by an instance of this random variable instead of the mean. So in this case, we consider that these postsynaptic response, these PSC amplitude or postsynaptic current amplitudes are gamma distributed. Why that is, there we argue in our paper. But essentially, we now have our linear nonlinear operation both for the mean and the standard deviation of this gamma distribution. And then each spike is sampled from this gamma distribution with mean mu and standard deviation sigma. So just to illustrate this briefly, if we have a kernel, here again, I take a monosynaptic mono-exponential decay as the mean kernel. And I take three synapses that have varying sigma kernels. So the mean response of each of those synapses is exactly the same. The efficacy at each spike is decreasing. But because of the shape of the sigma kernel, here the synapse in green will increase its variability, whereas this blue synapse will decrease its variability throughout the spike train. So we can capture heteroscedasticity that is often observed in, for example, patch clamp experiments with this model. I do think I have briefly time to show you this. another example of what we can do with atypical dynamics is that the shape of this kernel. So far, I've only shown you this kernel in terms of a exponential decay. But the kernel can be arbitrary. So there is this wonderful paper by Neubrandt and colleagues where they consider another synapse in the hippocampus from dentate gyrus granule cell onto interneurons. And they do a stimulation protocol in which a control spike is followed by a high frequency burst and then a test spike after a variable time interval. And what they show is this highly delayed facilitation where during the burst you have facilitation, but the actual EPSC amplitude only peaks 2 seconds after the burst is over. And we can capture such dynamics by just considering the shape of the efficacy kernel to be different. So here we take an efficacy kernel that is the sum of 3 Gaussians. And this...
if you can wrap it up in 1 or 2 sentences. Thanks.
Sure, sorry. Well, essentially, by varying the kernel and the baseline, we have a very flexible model here that we can use to describe a lot of these atypical dynamics. What I left out is that we have a method to infer parameters from experimental data and some more atypical plasticity dynamics in the paper. So if you want more, preprint link is in the chat. All the code is on GitHub. Please play around with it. Hit us up on Twitter. And if you want a real-life example of how to use this, in room 7, Michael's just getting ready for his talk on the Raphe nucleus. And with that, I just want to thank you and all my co-authors, and especially the Erasmus program, because they funded me during my master's to do this research in Canada. So thanks. And sorry for going over time.
Thanks for the wonderful talk. I'm clapping here for on behalf of the audience here. So my sense is that the audience are still typing questions and formulating it. So I'm just going to ask a really obvious clarification question. So we know that, as you also mentioned, right, one feature of the spike response plasticity models is that that they can make claims about their biophysical plausibility, right? And you can compare their parameters with measurements from in vitro patch clamp studies, for example. And so in a way, those models have a lot more constraints. Would you say this is why they have difficulty achieve the level of flexibility that your model can do with, different kind of kernels and stuff?
Yeah, I think that that's exactly the point. That if you have complex dynamics that are usually a combination of different factors that are interacting, if you consider a mechanistic approach, you need to essentially include a factor for each of those. And there are extensions to the Zurich-Markgraf model that can achieve that. It's just that you accumulate a compendium of parameters in the process.
Great, thanks. We will need to move on with the next speaker, but whenever there are questions that pop up in the Q&A window, you can still stay and ask the audience question in the chat.
Sure, thanks, Cheng.
Thank you. Our next speaker is Ganchao Wei from University of Connecticut. He will present his work on a very related note to the last talk, but instead of a mechanistic model, he will tell us a story more from a statistical point of view. So Ganchao, the stage is yours.
Okay, thank you. Can you see my screen?
Perfect.
Okay. So thank you for this opportunity. Basically today, I want to introduce my current talk with Dr. Ian Stevenson. That's the similar topic, tracking the fast and slow synaptic nerves by neural spiking data. Basically, now this is a synapse, and as I mentioned in Julian, there's a short-term plasticity, and which is a story about neurotransmitter release, and long-term plasticity, which is about the post-neuron receptor. And if we record the spiking activities for both pre and post-neuron and plot the cross-correlogram, we can detect some coupling patterns between them. This is one example from Swartz's lab. In this cross-correlogram, we can see that after some time delay, there's a peak here. That means the post-neuron fires nearly immediately after the firing of pre-neuron. And based on previous research, actually the coupling pattern or coupling weight will change along the time. And this changes occurs in different time scale. In the scale of millisecond, the short-term plasticity will give us the short-term changes. But for the scale of around minutes, things are more complicated. The long-term plasticity obviously will give us the long-term changes. But there's another very interesting but annoying long-term changes, which is actually caused by the short-term plasticity. This is now one example, also from Swartz's stuff. We can see there's a long-term fluctuations in pre-neurons, and we can also see the more pre-neuron fires, the less the probability of the post-neuron will fire. That means there's a short, the depression STP. So because there's a depression STP and long-term fluctuations in pre-neuron, you will produce long-term fluctuations in post-neuron. So basically, our goal is to use the spiking activities or spiking data to track both short-term changes and long-term changes. Also, as I mentioned, also want to distinguish this kind of long-term dynamics from the LTP. So to achieve these goals, we basically build a model based on the Poisson GRM. At each time TK, the equation is like this. The log link was used to capture the nonlinearity. And for the linear component, it basically can be divided into 3 parts: the long-term part, the short-term part, and the synaptic connection, which gives us the shape of the coupling effect. To fit the synaptic connection, basically we just need to fit the shape of the cross-correlogram by alpha function. So our focus now is to fit the long-term part and the short-term part, and how to combine these two. So let's focus on the long-term effects, which include the baseline and long-term plasticity. Here we choose to use a point-precise adaptive smoothing to handle this. It has 2 steps. The first step is filtering, and the second step is improve the estimation from the first step by backwardly smoothing. To do the filtering, we first need to define the evolution of the long-term effect. If we denote the long-term effects at time tk to be theta k, then and we assume it evolves like this with some Gaussian white noise. The basic idea for the filtering is that we first now predict at each time tk, we predict the parameter by this equation, and then correct our estimation by the real observation. And another thing I want to mention is that the pre-specified covariance Q will influence the estimation a lot. If we set it to be too small, the estimation will be too smooth. If we set it to be high, the estimation will be super noisy. But for short-term changes estimation, the smoothing can no longer be used because the STP is changing so fast. If we use smoothing, we can never detect anything. So the idea, I think, is very similar to the Julian. Basically, we handle it by additive effects that depends on the presynaptic interspike interval. In short, now we just convolve this modification function along the presynaptic spikes. What do I mean? Maybe take the depression STP, for example. This is a so-called modification function for STP, which is a function of presynaptic ISI. When there's a spike in preneuron, the STP will decrease sharply, and the amplitude is determined by this modification function. For example, now the ISI is small, which corresponds to the blue dot. And then after that, you decay. The effect decays exponentially with rate tires. And then if there's another new pre-spike, additive decrease and then exponentially decay, so on and so forth. So the basic question is how to handle this modification function. Here we just now choose to use a linear combination of basis function to capture the nonlinearity. So by doing this, the STP estimation just reduced to a linear model problem. Okay, so now we have method to deal with these 2 points separately, and we choose to combine them by alternating optimization. That means, for example, we first fix STP and estimate long-term pot, and in the next step, we fix the long-term pot and estimate STP, and then keep going on this process until the model events converge. So now we have the model like this, and we have the method to fit our model. The next step is to use some simulations to make sure that we can handle different situations. In those simulations, we play around different STP. All the baseline is assumed to be is generated as the constant, and the LTP is generated as stem changing at mid time point. And this column just show that we can handle it very successfully. Now we fix the STP to be depression and simulate data as linear change for the simulate the long-term plasticity as linear changing or sinusoidal changing. Things mostly looks good. But the linear changing and sinusoidal changing LTP is far from real situation. So how about the estimation for some more realistic LTP? In this simulation, we choose to use the STDP model to generate LTP. In that model, the LTP is assumed to depend on the presynaptic spiking time. And in this specific example, we use a double exponential function as a modification function for LTP, which is very common for cortical and hippocampal slices. Under this model, we can generate the LTP as shown in this black line, and things also looks good. But here, another thing I want to mention. Obviously, we can use the STDP model to model the LTP and estimate this kind of modification function directly. And actually, some previous research really did this. But the LTP, it's actually not depends more than the presynaptic spiking time. So here we just want to give an unbiased track for the LTP. How about the estimation on the different firing rate? Actually, the presynaptic firing rate will influence the estimation a lot. In those simulations, the baseline for the postsynaptic neuron is set to be constant. But we can see if the presynaptic firing rate is high, get a not accurate estimation for LTP. If it's low, then the LTP estimation will vary a lot. So basically, based on these simulations, we are more confident that our method can handle different situations. But there are some problems remaining to be answered. The first problem is that, do we really need to do all the things simultaneously? Can we just estimate them part by part, which will make our life easier? But unfortunately, we must do this. This just show one example. In the first column, everything was estimated simultaneously, and things looks good. But now, if we miss the baseline estimation, the fluctuations of baseline will flow and even enlarge in the LTP estimation. Similarly, if we miss the STP estimation, the fluctuation of the STP will flow into the LTP estimation. So in brief, if we miss any part of them, maybe the overall estimation will be good, but we'll get the spurious estimation in some other part. And also this plot just give an answer for the this goal. Yes, we can distinguish this kind of long-term, the long-term changes now induced by the STP from the LTP. Okay, that's the basic ideas for our current work. Are there any questions?
Thank you very much for the wonderful talk. And so we have one question from Danielle, who asks, how are you able to tell from extracellular recordings whether a cell is presynaptic to another?
Excuse me, can you, the recording? You mean the spike sorting problem, right? I'm not very sure, sorry.
Yeah, I think this is a more, I'm not sure about the question either. So basically here, your model is basically a statistical.
I see. I think now this question is more about the spike sorting problem. And in our model, we assume everything is not perfectly sorted now. We just know which spike belongs to which one.
Yeah, that's what I'm gonna say. So in the model, you would assume you know the perfect mapping. Like you are just assuming that you have two cells, you know them, which one is presynaptic, which one is postsynaptic. And
exactly.
It's possible to tell from the experiment is I think beyond the scope of this talk, right?
Yeah.
I mean, kind of. I know I'm really impressed how, like from your mathematical formalism, I can totally see how a point process adaptive smoothing, or is that what you call it, could produce long-term changes in synaptic weight. But I mean, whatever shape of long-term synaptic effects you would be able to reproduce. But I'm just wondering a little bit more about the possible biological mechanisms for this. I know Giuseppe is probably going to touch upon it later, but Ganchao, what would be your guess based on your work? So is it only, so does STDP completely, if you do a simulation with a network of neurons with only STDP mechanisms, would it capture the statistical predictions from your theoretical?
So you mean the multiple synapse case or multiple neuron case, right? Or a network of the...
Well, just maybe two neurons, presynaptic, postsynaptic. If you only assume, if you simulate their spike trains and you only impose STDP, would it reproduce? Would it match your expectations from your theoretical?
Yeah. Actually, now, if we know the true LTP or STP and turn off this estimation, we can obviously we can recover the estimation for any port. But now it's very hard now to distinguish those two ports. separately by experiment.
Okay, I have follow-up questions, but I think we need to move on. Thank you very much.
Sorry, thank you.
So next up we have Giuseppe Kindemi, who will tell us about his work with the Blue Brain project, which probably will answer the mechanistic questions about long-term plasticity. So I will yield the stage to Giuseppe.
Thank you very much, Cheng. So let me just share quickly my slides. Present. Okay, so basically the question that I'm trying to answer today in this talk is whether we can map the plasticity between pyramidal cells connections in the neocortex from the sparse data that we have. We know that the laminar architecture of the neocortex is one of its most prominent features, like this division in layers that is extremely stereotypical and reproduced everywhere in the neocortex. And we know that pyramidal cells are essentially the backbone of this architecture. The different types of pyramidal cells are those that build this. And plasticity of pyramidal connections is thought to be involved in learning and memory. However, only a few of these connection types, like for example, the layer 5 to layer 5 pyramidal cells, have been investigated in experiments. And so a complete map is still needed if we want to study learning and memory in the neocortex at a circuit level. So the objective of this work is essentially to try to fill the gap, generalizing what's known from the sparse data that we have, and to produce a first predictive map of LPP and LPD in the somatosensory cortex. The idea comes from a set of experiments by Sjöström and Neusser in 2006, where they show that the same SDP protocol that used in layer 5 pyramidal cells produce potentiation. So this is the very traditional SDP protocols that almost everybody is using, like several spikes paired at high frequency with a small delay between pre and postsynaptic spikes. Well, this very same protocol, if used in a different connection between pyramidal cells, specifically the layer 2-3 to layer 5, actually most often than not produce depression rather than potentiation. And what they show is that essentially the reason for this is that the location of the synapses determine that what's seen at this connection type is a very different calcium trace in the specific case. And so by boosting the backpropagating action potential in order to reach these distal synapses formed from layer 2-3 to layer 5 connections, you can actually recover some sort of potentiation. So essentially what they show here is that the differences in plasticity of these two connection types seems to arise from synapse location rather than different mechanisms. And so what we hypothesize here and we want to test is whether this is true for all connection types, not this very specific example, and if in general we can dump all the variability into calcium dynamics. And so by modeling calcium, we get for free plasticity of all the pyramidal connection types. This is our hypothesis. So how to test that? Well, first we started by designing a very basic model of synaptic plasticity, collecting essentially what's known in literature. Now the mechanism of this form of plasticity at pyramidal cells is well established. And the idea is that, essentially calcium influx into the spine from an NMDA receptor and calcium channels trigger plasticity mechanism that can in turn either change the density and efficacy of AMPA receptors at the postsynaptic density, or signal back to the presynaptic neuron to increase or decrease the release probability. And the way in which this happens, like a very common model of this, is the so-called calcium control hypothesis that state that intermediate level of calcium will most likely trigger depression, while high level of calcium will trigger potentiation. And so what we did, we took a model that essentially described this behavior from Graupner and Brunel 2012, represented here by this first equation that just describe synaptic efficacy, essentially, which is simply a variable that indicates between 0 and 1 if a synapse is potentiated or depressed. And basically, we plug this into a circuit model in order to get essentially these calcium trace that we actually need in order to be able to generalize. And then this model has been connected to the release probability. For example, in this specific case here, this is the Solis Markram model that was described before. So I won't go into the details of this, but just to give an example how this plasticity model can be connected into a synaptic change. As I was saying, in order to get the synaptic, the postsynaptic calcium trace, what we did, we plugged this model into the Blue Brain project microcircuit, which is a dense model of, in this specific case, of a cortical column that is built in order to match a large set of in vitro experiments ranging from connectivity to the electrophysiology and all the morphology of the cells and so on, in order to actually get a circuit that resemble and match as close as possible data that comes from the lab. After doing this, basically what we did, we parameterized this model in order to reproduce a set of experiments from Markram and colleagues in '97. And what we can see is that essentially our model can reproduce very well the results that from the in vitro experiments. Here, the open symbols are the in vitro experiments, the closed symbol is the model. And we can see that, for example, the traditional frequency dependence can be matched. The traditional STDP curve that is often described in papers can be matched. And also we find some nice emergent properties. For example, it was later shown in 2001 by Sjöström and colleagues that an LTD protocol actually gets converted into LTP if you increase the frequency too much. In other words, there is no LTD at high frequency. And this is something that emerged naturally in this model without really needing to fit these points, actually, because these points were really not there in the original datasets. In order to constrain the apical dendroid, we also use another set of experiments that is basically the one that I was describing before, the connection between layer 2-3 to layer 5 pyramidal cells, and this specific point that I was showing you in the early slides. And also here we can see that in this connection type, we more or less match the in vitro point, or at least we are much closer to the in vitro point that we have for layer 2-3 connection than the one for layer 5, showing that indeed the model predicts a certain difference between those two. And also we can repeat the very same simulation protocols that we did before to characterize what's going to happen for points that we actually don't have. And one nice thing we see, for example, here for layer 2-3 to layer 5, our model seems to indicate that we don't really see an STDP curve anywhere. Also, you can see here, this is just a summary of all the frequencies and DTs that we tried. And so this is a prediction that needs to be tested then experimented. But now the funny part begins, because all these essentially connection types is something that we use for fitting. So what this shows is that indeed it's possible to fit a single model that captures two different types of pyramidal cells without really needing a specific parameter set for each of them. Actually, here all the variability is dumped again into the calcium trace. It's simply that these synapses are at different locations, they have different physiology, so their calcium trace is different inherently. So now the funny part begins, because we can really test the same approach on connections that were not used for fitting in any way, they were not there at all. And, but yeah, I forgot to mention that another important part of this set of experiments is that this model seems to reproduce also the distance-dependent plasticity effect that Sjöström and Heuser modeled in their original set. But I mean, here the interesting part is actually what comes later when we test the model on PC connection that were never seen in training, not even the same type. For example, when we tried layer 4 to layer 2-3 pyramidal cells, here we can see the match is almost perfect. And here actually the black symbol is behind the white one. I may apologize, it's just a very good match. And we could even do a little bit of pharmacology here. For example, the triangles here match essentially experiments that were carried on, including MK801, which is an NMDA blocker, into the presynaptic pipette. And here, by simply decreasing the amount of depression, so blocking essentially, mimicking in some sense the effect of blocking presynaptic and NMDA receptors in this set of experiments, we could actually reproduce quantitatively the results of this protocol. And again, the same seems to hold true also for layer 2-3 to layer 2-3 connections, where qualitatively, although here quantitatively is a bit different, we still get that 20 Hertz stimulation protocol seems to induce potentiation at this connection type. Here we get a little bit more than what was recorded in experiments. But again, this is a completely new set of experiments. It's something that has never been seen during training. So this is actually an encouraging result. Now it's interesting also to see where the model breaks. And here, for example, we try to do the same approach on a set of connections between spiny stellate cells. Spiny stellate cells are still excitatory cells, but they're non-pyramidal. So it's a different type of cell. And as we can see here, our model simply does not behave well. And this was kind of expected because what the Egger and al. Showed in this set of experiments is that indeed the plasticity of this connection type is non-NMDA dependent. It's not really the type of plasticity that we would expect from pyramidal cells. So actually, it's in some sense also fair that the model breaks here because it was never meant to be a model of all excitatory cells. It's really something specific to pyramidal cells. And also, one nice thing of having such a model is that you can also not only play with different connection types, but you can also play with different conditions. So one thing that we tried to test is whether by dropping calcium, as we also saw in a few talks before, if actually the plasticity of this connection seems to change. Because one thing that is often neglected is that calcium in vivo is actually very different from calcium used in vitro experiments. In vitro, given that the slices don't have lack most of the connectivity and everything, in order to awake, let's say, as often said, the slice, usually people increase calcium to 2 millimolar, extracellular calcium to be 2 millimolar. But in vivo, usually levels are much lower, ranging between 1 and 1.2 millimolar. And so what we see here is that indeed the landscape of plasticity, it's very different when you do this. And for example, here, the traditional 10 Hertz STDP curve here is completely lost. If anything, we get these STDP experiments at much higher frequencies. And in general, overall, the magnitude of these is heavily reduced. So it's really a different thing doing these kind of experiments in vivo. And these has also recently been shown in some in vitro experiments from Brunel and colleagues. To conclude, I just wanted to say that, so this experiment seems to point in the direction that pyramidal cells share a large set of plastic mechanisms overall, and so the heterogeneity that we see in experiments could be really due to differences in calcium rather than in plasticity mechanisms. And if this is true, then this means that only a few targeted experiments could be necessary for us to really capture the plasticity of pyramidal connections in neocortex. And last, that in vivo calcium is something that shouldn't be neglected when we want to conclude about plasticity from slice experiments, because indeed the situation in vivo might be drastically different. And with this, I finished. So thank you very much. If you want to have a look at this paper, we have it in bioRxiv for the moment. So please feel free to have a look, and thank you very much. And if you have questions, of course, I'm more than happy.
Thank you, Giuseppe. We have one question from Yuri Rodriguez. Said, so the sparse data we have to reconstruct the plasticity rules are produced in very different experimental conditions.
Yeah.
How are modelers dealing with experiments made in different temperatures, animal developmental stage,
essentially
compositions, etc.?
Well, essentially, you just go crazy because like you have to compensate for all these effects, and these drastically reduce the amount of data that you can use. Here, as you can see, I didn't rely or I didn't use a lot of experiments, mostly because in order to match exactly what you were asking in this question, the number of experiments that you get is heavily reduced. For example, here I had to exclude all the experiments done with extracellular electrodes, because in that case, we don't know who is the presynaptic partner. So we have no chance of actually doing it specific for a connection type. Then also all these experiments, or whenever possible, come from the somatosensory cortex, because the circuit model that we were using is a somatosensory cortex. In a few instances, I had to use visual cortex, but that's of course not ideal, because visual cortex is a different system in the end, and also in terms of plasticity, it seems to behave differently, quantitatively at least. Then temperature, same thing. Like you try to rely as much as possible on data done at physiological temperature whenever it's available. Calcium is another thing. It's just drastically reduce the amount of data that you can use. And one has to be fair and just exclude those points that it cannot really account for, I would say.
Yeah, thank you, Giuseppe. You did a bunch of really comprehensive studies. I'm really impressed with the common set of plasticity mechanisms among pyramidal cells. I wouldn't have expected this at all if I didn't know about all of these data. So I think we are at time to conclude this session. And I really enjoyed talking to the three of you. And I also appreciate the discussions and the attention from the audience. And the next bunch of sessions will start in 12 minutes. So you guys will all have time to grab a cup of coffee or something. All right. Thank you very much again, and goodbye.
Thank you.
Thank you for sharing. Cheers.
Bye-bye. Thank you.
But it was broadcasting.
Even when we're in the green room?
Mm-hmm.
That's really weird. So Mark, we're going to be on YouTube. Yeah.
Exciting.
So is there a way to have your Zoom room for video not showing? I think that's a setting.
I think it doesn't show, but I normally press like the button to view only the attendees, the panelists that have activated video.
Right. All right. So let me hop over into the.
Okay, we go in 1 minute.
So Mark, I'm going to keep it pretty brief.
Please.
Are you a capital P professor? You are, right?
Yeah.
Yep.
Yeah, briefer the better. I did a practice and it was like an hour and a half. So.
Yeah, the slides are currently showing up on YouTube.
I start broadcasting.
I'm going to keep it pretty brief.
Please.
Whoa.
Are you?
Yeah.
Are you okay?
Turn that down. So the YouTube is delayed by like a bit, and only Mark is showing up. That's good. Okay. Hello, everyone, and welcome to this session. It's my tremendous pleasure to introduce a good friend of mine, Mark Howard, who's a professor at in the psychology department at Boston University, and he's going to tell us about some of the work he's been doing lately to understand how the brain uses time cells to think, represent space, time, and memory. So without further ado, I'm going to turn it over to our distinguished keynote speaker. Go ahead.
Thanks, Brad, and thanks to everyone who's worked on organizing this online conference. It's really exciting to see people sort of reinventing ways of how we do business as academic scientists, and it's really great. So go team. So I have a confession to make, and that is that I'm not really a neuroscientist. I started in cognitive psychology, and I've worked in mathematical psychology and cognitive science, and I've sort of come into neuroscience sort of necessity. And the reason I've come into neuroscience is an attempt to place physical constraints on theories of how we might think and remember and do things. And in this talk, everything I'm going to show you is going to be motivated by this theoretical perspective, but I'm not really going to be able to go into that in any detail in this talk. So this might seem a little nuts. But I encourage you to look into other work that we've done, as that's provided inspiration and background for this. And I'll allude to some of this as we go along. So about 10 years ago, inspired by ideas from like really the dawn of psychology, William James, for instance, talked about this a great deal. My student Kartik Shankar and I worked out a hypothesis for how the brain might maintain information about the recent past and how that information might be used to construct cognitive models of a variety of memory tasks. And here's the basic zero equation hypothesis. So as an event happens, as something unfolds over time, we listen to, for instance, a melody, boop, 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 something like that. As it recedes into the past, neurons in our brain actively maintain a record of that recent experience. And they, much like the musical staff going diagonally in this figure. And so that at each moment, we have a record of what happened when in the recent past, and that different neurons ought to be responsible for different parts of the past, so that our memory is organized like a timeline of recent experience. And so moreover, in addition, very much inspired by, again, very long-standing ideas in cognitive psychology, we proposed that the resolution of this temporal axis ought to be compressed, such that you have a less good idea of when things happened further in the past than more recently. That we have a good memory for the recent past and more fuzzier information about when things happened in the past. And so you can operationalize this as the brain contains a population of neurons with receptive fields organized along a time axis, right? And one population of neurons is responsible for things that happened maybe 1 second ago, and another population is responsible for things that happened 2 seconds ago, or 4 seconds ago, or 8 seconds ago. Okay, so that's sort of where we started. Around the time we were making this proposal, people discovered a phenomenon called time cells, and people here mostly Yuri Bushaki's lab, Eva Pashtelkova in Yuri Bushaki's lab, and then Chris McDonald in Howard Eichenbaum's lab, and the great Howard Eichenbaum did a number of really important studies on this. But they observed a population of cells that behave kind of like what we were looking for. And so let's think for a moment and think about how a population with receptive fields in time would behave as something as an event recedes in the past. So if some triggering stimulus comes, clap, as it recedes into the past, it should enter and then leave the receptive fields of different neurons, different time cells. And so if we're observing from the outside, what we should see is neurons that fire in sequence, that fire at a consistent sequence across different occurrences of the same stimulus. And on the left here, I'm showing you three very nicely chosen hippocampal time cells from dorsal CA1. And here I can show you a population also from the Eichenbaum lab, a population of 172 simultaneously recorded neurons sorted on their peak time. Okay. And so this population, I hope you can convince yourself of quite quickly, carries information about when the stimulus that started this sequence began. Right? By noting which cell fires, I can figure out how far in the past that thing was. I'm going to tell you about a couple of more results from work that from various work showing that different stimuli can trigger different sequences, such that if we have a boop and a beep, we'd get different sequences of time cells. And so by noticing which cells are firing at any particular moment, we can reconstruct not only when the stimulus happened, but also its identity. I'm going to argue that the time axis, as we proposed a while ago, is compressed such that it doesn't have equal accuracy for different parts of the temporal past. I'm going to argue it's probably a logarithmic compression, although you could fight about that. You can actually appreciate really rapidly that the time axis ought to be compressed to the extent that you appreciate that there's a curvature here in this population of cells. If the receptive fields were evenly spaced on the time axis and we sorted them we started the sequence on at peak time, we'd see a straight line. The fact that we see a curved line like we do in this figure means that there's many more cells with receptive fields at the recent past and fewer as they go along. And we'll formalize that in a minute. And I'll also emphasize, although this phenomenon has received a lot of attention in the hippocampus, which is presumably responsible for episodic memory, we see kind of the same phenomena. We see very similar phenomenon in a number of other brain regions that are prefrontal cortex and striatum that are presumably responsible for working memory and implicit memory or conditioning, however you want to define that. Okay, so I'm going to go through a series of studies here and sort of convince you of these points in more detail. This is a paper from recordings. Recordings were done by Earl Miller's lab, originally published in a 2010 Neuron paper. We're looking here at reanalyses by Zaran Tiganj, who's currently in the Department of Computer Science at Indiana University. And in the task, the monkey has to remember what stimulus began the delay. So it's a working memory task, and the animal gets either a dog or a cat, a picture of either a dog or a cat, and then they have to wait, and they get another picture, and they have to say whether it was the same category, yes or no. And there's two categories. There's two types of categories. There's cats and dogs, and then there's cars and trucks. And to the extent we can figure out, to the extent we can decode which stimulus was at the beginning of the trial from looking at the firing rate during the delay, we can conclude that this population has information about what happened when, much like the different notes on the staff and the figure I showed earlier. So the first thing I want to establish is that there's time cells in this data set. I'm showing you a plot of the active cells during the delay interval as a function of time, and hopefully this looks to you similar to the previous such plots I showed with time cells before. But moreover, the activity depends on not only when the stimulus happened, but also the identity. And so in order to evaluate this, what we did was we took each neuron and we sorted it on the basis of its best stimulus. So let's say the best stimulus is a dog. And then we compared the firing for the same cells with the same sort to the other stimulus from the same category. So if that was dogs, dogs are the best. And then this is cats, also from the same type of domain. We see kind of a similar temporal curvature. However, the same cells fire much, much less to cars and trucks. So we can see, meaning that cells respond best to one particular type of stimulus, and their firing gives us information about identity within the category as well as the different categories, such that we can look at firing during the delay interval and construct a decoder and decode well above chance whether this trial began with a cat or a dog or a car or a truck. And in this analysis, and many people have done this, so hopefully this is familiar, we trained a classifier on a segment of time, say a 50 millisecond bin, say 0.75 seconds after the stimulus, and then we tested that decoder on each of the other time points, and we find that the decoder is above chance, indicating that that the population has information about what happened, but also it decorrelates over time, and that's a consequence of the fact that there's sequences of cells, that there's also information about time in this population. And so I chose this study because we had it handy, and it's especially nice, but we've also seen the same results from monkey PFC and hippocampus. Nathan Cruzado has a paper, should be out in Hippocampus briefly, that uses the same type of analysis on a different kind of task. And other groups have seen this as well. Yanis Tashidis from Payman Golshani's lab just recently published a neuron paper showing the same type of thing from rodent hippocampus during perception of odors. Okay, so it seems that this population of time cells carries information not only about when the triggering stimulus took place, but also when it took place, as we propose. Okay, so I want to talk for a minute about the compression hypothesis. Actually, let me go over to here. So we here, meaning neuroscientists, have a lot of evidence that the brain represents continua out in the world along a logarithmic scale, okay? What I'm showing you here is a very famous set of figures from the visual system, where we're plotting size of visual receptive fields on the y-axis as a function of distance from the fovea on the x-axis. And you see that rather than cells throughout the visual system rather than receptive fields throughout the visual system having a same characteristic size, they get wider as you go to the periphery. And this has been understood for a very long time. And you see a cartoon down below. It's also been known for quite some time, dating back to the 80s, it seems that we resolved this pretty well, that the compression of the visual system, at least outside of the fovea, seems to be as if the receptive fields are evenly spaced on a logarithmic axis. So let me go back to this other picture. So if we had receptive fields that were evenly spaced and evenly tiled a logarithmic axis, we would see when we looked at them as a function of not log, there's the regular retinal position, we would see that as we go away, the receptive fields get wider and less numerous, right? And we believe that this phenomenon, this logarithmic compression, supports, at least in some circumstances, the Weber-Fechner law, which is a very, very old result from psychology, and holds for nonverbal numerosity as well as vision. And we proposed that this ought to be this same sort of compression ought to hold for time as well. Okay, so we're going to try and evaluate that. And so here I'm showing you some preliminary results from recordings by Jay Bladen in the Eichenbaum lab with analyses being done by Ray Tao in my lab. And what Ray did was she estimated, first of all, you observed time cells in this experiment, as one would expect. What she did was very smart. She estimated the receptive fields rather than averaging across trials. She averaged the receptive field on each trial and estimated the distribution of locations using hierarchical Bayesian analysis. Okay, and so you can see 3 example cells, much like the ones I showed it earlier from Chris McDonald, with this fit taking place, and you can see the little squiggly lines. The width of the receptive field is now estimated within trial, and we can separate that out from the across trial variability. And if there's logarithmic compression of this time axis, we would expect a linear relationship between that within trial receptive field width and the location across trials. And C here shows the width of the within trial receptive field as a function of its location. And to the extent you accept that's a straight line, you accept that there's logarithmic compression. She also estimated the number density of cells. So I've been saying for a while that this curvature is consistent with the compression. It turns out that a logarithmic compression has a very specific form, and we should be able to express, if there is literally a logarithmic compression of the timeline, we should see that the number density is coming from a power law distribution with an exponent of 1.0, and she estimated. She fit the distribution of means across cells, again using hierarchical analysis, using a hierarchically constrained analysis. And the inset here on the left shows, and hopefully you can see it, shows the posterior distribution of that alpha, and it overlaps, it lands exactly on 1.0. The little vertical line hits 1.0, so that's a good job. So it seems that we certainly can't falsify the hypothesis that the compression is logarithmic, and there's strong theoretical reasons to think that ought to hold as well. So let me sum up what I told you about time cells just now, and then I'll tell you about some other stuff, which is also really cool. So time cells fire in sequence, and they carry information about the time since the event that triggered this sequence, okay? We know that different stimuli trigger different sequences, carrying information about the stimulus, the event that triggered the sequence. Because of these 2 properties, we can decode what happened when from this population. we're certain that this the temporal axis is compressed. It's not, there's decreasing accuracy for events further in the past, and we think it's probably logarithmic. And I would also add that I showed you results from hippocampus and prefrontal cortex. Other people have observed similar phenomena from the striatum, and that was also part of our original proposal, that this memory for the past, this sort of experience of the recent past with what happened when, ought to be important in many different forms of memory, including working memory and episodic memory and conditioning as well. And there's pretty good arguments for that that I can't go into today. Okie doke. All right. So that was part of the stuff we wrote on the chalkboard 10 years ago. We spent a while trying to figure out how you could generate a population of cells that coded for the past with this logarithmic compression. And we could only think of one thing. And once we thought of that, it seemed like a really good idea. And so we've pursued this for a while. And I'm going to tell you basically about recent evidence for this other kind of coding. So we proposed in the 2010 paper and subsequent stuff as well, that there's not just time cells. The time cells are derived from this other population of cells. Let's call them temporal context cells. And rather than having receptive fields that tile the time axis with different having different peaks along different parts of the axis, we proposed that these cells ought to code for the same type of information with exponential receptive fields, and that rather than having different time points, rather than peaking at different points like those Gaussians I moved along, that the variability across cells should be carried by the rate at which they decay. Okay, and so here are temporal receptive fields for as a stimulus is a certain amount of time in the past. One cell might have a fast time constant, and other cells might have slower and slower time constants. But there's this family of receptive fields that have exponential decay that would also be used to generate time cells. Okay, that carry information about the past. This is also the real Laplace transform of the past. If you had a population of cells with spectrum of rate constants, the reason we know they would be coding information about the past is they're the real Laplace transform. But we can omit that for a moment. Let me try and take a moment and sort of convince you that a population of exponentially decaying cells with different rate constants S you'd be able to figure out when a triggering stimulus took place using that population. So first, I'm showing us our old friend, an exponential curve. I've chosen the rate constant S and the time constant 1 over S or tau to be equal to 1, and this is an exponential curve that hopefully everyone's seen a bazillion times. Okay, if we zoomed in on this function, so if we looked at times much shorter than 1, we would see that this cell is not conveying useful information about that time. I've just taken exactly the graph I just showed you and zoomed way in on the x-axis. Similarly, if instead of zooming in, if I was looking at times much larger than 1 second, like so, and I've just taken exactly the same curve and zoomed way out, the cell is also not conveying useful information about the time at which that event took place. The firing for time 100 and time 150 and time 200 and time 250 is different for this cell. It's identical. What the cell is doing for you, though, is telling you good information around the time scale of its time constant. And basically what it's doing is giving you the ability to tell whether the triggering event was earlier or later than its time constant. So if I had a lot of cells, here I've drawn, hopefully you can see this, 3 cells with 3 different time constants, red cell, blue cell, and green cell. If I look at this population and I'm able to simultaneously observe all of the cells, and I find that, for instance, red cell has turned off, but blue cell has not, I know that the time of the event must have been somewhere between the two time constants of those cells. Similarly, if I observe that green cell is still firing, but red cell and blue cell have turned off, I can figure out that the event was somewhere between the time constant of blue cell and green cell. And by having many cells turning off in sequence, I can figure out how far in the past the stimulus took place. There's like an edge as a function of time constant of these cells that I can use to infer how far in the past the triggering event took place. And that's the basic idea. More formally, we can, and this is sort of where we started with, more formally, we can say that a population of cells with exponential receptive fields, that's all I'm showing you right here, f of s, is just a population of cells with a set of exponential receptive fields with different values of s, being convolved with being activated by the actual history, taking into account the actual history. That's the real Laplace transform of f of t. So the real past is related to the Laplace transform in a one-to-one relationship. And we can know, much like a distorted mirror carries information about the image, but need not look exactly like the image. That's my excuse for showing you a picture of Mo Howard. In addition, we know how to invert the Laplace transform, so we can imagine a different set of cells constructed by an attempt to invert the Laplace transform, and then it turns out they would look like time cells. Right? So the idea is that time cells behave like an attempt to invert the Laplace transform, and that there's this other population that codes for the transform. And so to summarize, for the last decade or so, we've proposed the following theoretical framework where some event happens, click, some f of t happens at some point in time, a bunch of cells activate at about the same time and then turn off at different rates. And that's used to generate a set of basically time cells that have receptive fields in time that are compressed. And I'm just in the middle column here, I'm showing you the heat maps you'd expect from a population of such cells. And I've chosen, and we've thus far seen a ton of evidence in this talk and also elsewhere for populations of cells that look like these attempts to invert the Laplace transform. But until quite recently, there wasn't evidence for this other population holding the Laplace transform. Okie doke. So, but that has changed. Over the last 2 years, we've seen quite strong evidence for this. I'm going to focus on a paper that's out in PNAS over the summer from students Ian Bright, Miriam Meister, and Nathan Cruzado, reanalyzing data collected by Miriam in Beth Buffalo's lab. And the monkey we're recording from the entorhinal cortex, and the monkey just sees a picture every so often. And we're syncing to the time at which the picture is presented, and that's it. And so a bunch of pictures are presented. And I'm showing you 3 cells that, rather than firing sequentially, all change their firing. Most of them are excited, but there's some that are inhibited at around the time the image is presented. And then they relax back to baseline with at different rates, with different time constants. Okay. And so here is a plot of all of those cells sorted like one would, sorted in much the same way one would for time cells. And rather than seeing this J that tiles the interval, we see essentially a vertical strip, because all of the cells across the population activated about the same time, and then they deactivate at different rates. And for funsies, I'll just remind us what the model picture would look like, and then quickly go ahead and you can convince yourself that does or doesn't look the same. Okay. We can also decode time, and we did a bunch of methods and stuff, and you're welcome to, happy to look at the paper, and I'm happy to answer questions about in the Q&A. And also we know that you can decode time from this population. If we ask, if we train a decoder to try and guess how far in the past the stimulus took place, we can figure that out. And this is a temporal decoder. To the extent there's a bright line down the center, you're convinced that the decoder is able to decode time. All right. So let me take a moment So thus far, and there's a paper by Albert Sau in the Moser lab showing essentially the same results for rodent LEC from 2018 in Nature, and so we're pretty excited that that was confirmed. And so far, we've basically caught up to the theoretical proposal from a decade ago. In the last decade, since we wrote that stuff down, we've continued thinking about cognitive science and cognitive psychology and computational stuff and why this might be useful. And I at least have become convinced that this is a really useful idea for like a theory of brain cognition. And I don't have time to convince you of that in this talk, but I'm taking this very seriously. And I don't think you're doing a bad thing if you also take that seriously. To give you an opportunity to try and take that seriously as a model of many things, I'm going to show you really super quick a study on a completely different domain. So this is Rifki Afan reanalyzing data that's previously published from Carol Svoboda's lab, 2018 Journal of Neuroscience, Inagaki. He's the first author. And this is a motor preparatory task. And what happens in the experiment is the rodent gets a tone, boop or beep, and then they have to make a response later. They have to withhold a response for some period of time. It's an easy decision. If you get the low tone, you're rewarded for licking to the right. And if you get the high tone, you're rewarded for licking to the left. But you can't respond yet. You have to wait. So rather than like a beep receding into the past, there's this planned action of a future lick that's at some point in the future that comes towards you over time. Right? You have to plan on being able to make this lick at a particular moment in the future, and you have to wait. And what these authors and other people have observed is that during the interval where the animal's waiting, these cells in ALM, anterior lateral motor cortex, ramp up over time as if they're growing towards the bound. As if they're ramping to the time at which the motor plan is going to be executed. And you can decode well above chance which, I think this is what I'm showing you. You can decode well above chance which movement the animal is going to make. Whoops, oh no. So what Rifki did was he took the analytic tools that we developed for entorhinal cortex and just applied it to this data set, which is very generously shared on crcns.org. And if you have a set of cells coding for the time since something happened, I'm just recapitulating results from the entorhinal cortex on the left here, you should see this pattern like so. In contrast, if you have a population that's coding for the Laplace transform of the time until something is going to happen, right? It should look like a mirror image of the results that we saw from entorhinal cortex. And I've shown you in the examples I showed you ramping cells that had a variety of time constants. Here I'm showing you sort of basically the same idea. To the extent you accept that there's a mirror image between the left side of this plot and the right side of this plot associated with the change in sign of the remembered time. Here it's positive, here it's negative. You accept that this population holds the Laplace transform of the future. So that's work in progress, but I think it's really unlikely that this would have worked as well as it did unless there was something to this idea that the Laplace transform is good for many things. All right. So to summarize, we have, I've shown you results where time cells in the hippocampus estimate functions over the recent past. They're compressed, etc. I've also shown you results that these temporal context cells in entorhinal cortex behave as if they're estimating the Laplace transform, real Laplace transform of the objective past as functions of time. I also showed you some preliminary results suggesting that this Laplace inverse pairing. Well, first of all, the fact that the hippocampus looks like the inverse and the entorhinal cortex looks like the transform, that places really strong constraints on where time cells come from. Time cells receive input from a Laplace transform of the recent past and behave as if they're an attempt to invert the Laplace transform. I've also showed you evidence that other brain regions, in this case ALM, at least use this idea of Laplace, behave as if they're using this idea of Laplace transform to code for other variables. Okay. And so to summarize this whole thing, I think this hangs together really well. I haven't really talked much about the psychology, and I haven't really talked much about the theory, but there's really good reasons to think that this Laplace transform is extraordinarily useful computationally. There's a paper by me and Mike Halsamo that's on archive right now that I encourage people to take a look at if they're interested in getting a high-level overview of these ideas. This is really general, and I'm quite excited. Here's people whose work I highlighted today. Zoran Tiganj is currently a faculty member at Indiana University. Ray is a postdoc in my lab. Jay Bladen is currently used to work with Howard Eichenbaum, is currently a postdoc at Brandeis with Shantanu Jadhav. Steve Czerzinski is applying to graduate school. And so if you're looking for a PhD student, Stephen might be the one you want. Ian Bright, Miriam Meister took the co-lead on that PNAS paper. Nathan Cruzado, who will be graduating in May, in case you're looking for a postdoc, is contributed to that paper and a number of other things, and has a special expertise in this cross-temporal decoding analysis. And Rifki Afan is actually a student in Ben Scott's lab, who's done this really remarkable preliminary work on ALM. Kartik Shankar, this whole Laplace thing was Kartik's idea, and that's a picture of Kartik Shankar, and good job, Kartik. And here's back when we had lab meetings, picture of the lab. I'd also like to thank my faculty collaborators, Mike Halsamo, Howard Eichenbaum, Earl Miller, and Beth Buffalo. I showed you work from them today, and of course funding agencies. And so thank you very much. All right.
Thank you, Mark. That was fantastic to hear about. And it's really amazing the way that the empirical and the theoretical perspectives on this have come together. It seems one of those fairy tale things in science. It's just really great.
I remember you being really excited about this back then, and you were much more excited than me, actually. I didn't think this was going to work as well as it did.
Yeah, I mean, I love time. I study time and attention. It's been underemphasized, I think, in terms of relative to spatial information, but it's really crucial. We've got a lot of great questions that I think are going to chew up easily the next 30 minutes if we let it. So let me jump right into it. The first one is by Nadine Spichala. And the question is this: is it a good idea to call these time cells? They probably carry way more information than just about time. Or is there any sensitivity of these cells with regard to time as opposed to other things they could also be sensitive to?
Yeah, I think, so we have to call them something. I don't think there's such a thing as place cells. I don't think there's such a thing as time cells. I don't think there's such a thing as splitter cells or any of the other types of cells we talk about. The verbal label is a shorthand that we use to be able to talk to each other, so we know what phenomenon we're talking about. I didn't talk about it here, but theoretically, we can construct place cells from the same Laplace transform inverse framework as we use for time cells, right? And we can also imagine a continuity of dependencies in between space and time. Howard and I did a paper in 2015 about this. So yeah, it's a useful verbal convention, but please don't take anything more seriously from that.
But these cells are primarily representing time rather than, for example, featural information about specific events like this light, that square, et cetera.
I think, well, but they also represent that too, okay. Right? Time cells are conjunctive with position, right? In Chris's original paper, if the animal moves around during the delay, you see cells that fire only when he's in a particular position at a particular time. So in the same way, there's conjunctions for what happened when, there's also conjunctions for what happened where and what happened where and when. And there's conjunctions of absolutely everything in the hippocampus and the prefrontal cortex. And it's just a verbal convention as to what we choose to focus on. Happily, this Laplace transform framework is pretty general, and we can apply it to many combinations of variables, many variables, and also many combinations of variables. Hello.
So next question, that was a great one, thank you, is by Matthias Hennig, who asks, would you have suggestions or hypotheses about how long these much longer than the neural time constants may be achieved by the single neurons in these recordings?
Yeah, so Mike Hasselmo and I did a couple of papers about this. First of all, there's a number of different mechanisms. You might imagine that there's recurrent connections with eigenvalues close to 1, right? And you also might imagine that there's intracellular processes based on, like, for instance, ICANN currents that are triggered by acetylcholine. There is such a thing. Angel Alonso did pioneering work on that going back like 2 decades. And Mike Hasselmo is an expert in that. For me, I'm kind of agnostic about the way you build out these equations. I'm really a cognitive psychologist. I think the question of how to build these equations is something that computational and systems neuroscientists should answer more than me. I will say merely that there's a number of different ways it could happen, and I'm partial to the ICANN current story.
Great. Next question is by Jules Brochard, who asks, is the time compressing on slide 5 a kind of efficient encoding?
Oh, yes, absolutely. And that's why we've felt quite strongly about this logarithmic compression for 2 reasons. One is, the analogy with vision and numerosity. But the other question is, why is this true for vision and numerosity? And the answer is that must be something, right? Why is this a common principle throughout the brain? And we've done some work. Kartik and I, and me and Kartik, wrote a couple papers about this a while ago. And the best answer I can give you in a soundbite is basically that because the brain needs to have information about a number of different scales, and it doesn't know a priori what scales are going to be important, right? So if I have A and it predicts some outcome in the future, I don't know if that's going to be 100 milliseconds in the future or like 100 seconds in the future. In order for me to equalize the error that I have for all of those possible scales without placing a strong prior on my expectation for what that scale would be, you end up coming out with a logarithmic compression. And there's a Psych Review paper from 2018 where we formalized this a little bit. I would also point out that there's a paper up on arXiv by Alexander Shu and Sarah Marzan making a more formal argument that I encourage you to have a look at, rather than me trying to remember everything in the paper.
All right, next up is another great question I'm sure a lot of people are thinking about, is whether these time cells would also work for keeping track of long time intervals, like 1 or 2 hours, or even further. Like, what's the sort of longest range that you think?
Yeah. So first of all, we don't know what the longest range is thus far because nobody's observed the sequence stopping, right? We've observed time cell sequences going on as long as the delay intervals in different experiments. We have some results, and my student Yue Liu, who's now a postdoc at NYU with XJ Wang, we think we see sequences going on for 10 minutes, but there need not, there's no way on earth you can have activity-dependent firing that lasts for days. Properties of human memory have the same, human memory has the same properties over every scale we can measure, from hundreds of milliseconds up to tens of minutes, up to days and weeks and months. There's no question that there has to be a different type of neurobiological mechanism to go over such long time scales. And we have some thoughts about this, but you can have thoughts too. Our thoughts aren't necessarily that great.
Are you able to, I mean, I guess one of the limitations is that as you get further back in time, the logarithmic compression means that you would basically need more neurons recorded simultaneously to get evidence for that.
Yeah, that's part of it. But I also, and Mike Hasselmo has said this forever, at some point you go to sleep and, the firing changes, right? So I'm relatively confident that you could have a continuity of scales up to maybe half an hour or 40 minutes, something on the order of an experimental session. I think that's reasonable. Things that happen over a longer time scale, you have to appeal to something else. And you have to have something like changing synaptic weights that decay at a variety of rates might be a way to do it. Or have memories recur such that you recycle memories as you go through your day, such that different parts of the brain sample at memories at different timescales or something like that, so that you get this gradient. The equations will work no matter what. So the question amounts to, how do I build a spectrum of timescales? How do I build a bunch of different S's where the time constants go up to whatever upper limit of the scale you're interested in?
Right. I mean, you could imagine though that even like out to 10 minutes as you observe these time cells experimentally, that the firing of those neurons is perhaps being supported by completely different mechanisms, right? Like within a few seconds, it's like active representation, and then you kind of shift over to like a synaptic mechanism, maybe, yeah.
I will say that regardless of the mechanism that causes the activity-dependent firing, the cell paper from the Moser lab, they showed exponential decays with time constants out to like an hour, right? So somehow, and it need not be the mechanism we've proposed, but somehow there manages to be activity that extends over many tens of minutes in the lateral entorhinal cortex of the rodent while he's going about his day, that codes for what happened when. And this in that experiment, it's when did the animal enter a particular enclosure. You can decode that out to very long periods of time.
Do you have a specific proposal about the EC subregions? So the question here is, do you assume that the temporal context cells are, do you assume that the temporal context cells are specific to the LEC and its primate homologs? How do reports of time cells in the MEC fit into this picture?
Yeah, I think what we should I think this is a really good question. I think there hasn't been a systematic mapping. In order for me to answer that question, I'd want to know to what extent there's a spectrum of timescales and spectrum of spatial scales throughout these regions. Like, we know there's border cells which behave as if they're Laplace transform of distance to an edge in the environment. We don't know if they have different space constants. That's the first question I'd want to know. We see in preliminary work, looking at data from the Halsamo lab, we see that grid cells are conjunctive with head direction cells, are conjunctive with speed modulation, but that the time constant of the speed modulation has a spectrum. So I think really we need to do careful experimentation with this stuff in mind to try and piece that out. The part I feel strongly about is that there's conjunctions and many variables. How those get assigned to different parts of the entorhinal cortex or the hippocampus or what have you. There's nothing the equations tell us about that.
We got time for maybe a few more questions here. So what is the probability that a neuron representing, for example, t=1 also fires at t=10? Can a logarithmic distribution fall out of this variability without neurons that are explicitly t=10 neurons?
Say this again.
So I guess the question is whether or not this pattern of time cells could arise spontaneously from a probability distribution of the way that neurons fire with respect to time. So imagine, I'm trying to picture this in my head.
Okay. So so the bit about how well, first of all, they're consistent across trials. So whatever they're doing, it's consistent from one trial to the next. And whatever they're doing, it's sensitive to not only the time at which the trial began. And so you remember the pictures with the rasters that are lined across trials, and they jitter around a little bit, but they're kind of in the same spot. And they also care about the identity of the stimulus that triggered them. So, I mean, in some sense, a spike is an attempt to is carrying some information about what happened when in the past. In some sense, you can think of that as sampling from a probability distribution. But the point is that the probability distribution depends on what happened when. It's possible there's some subtlety about the question that that I didn't gather.
But the fact that they're consistent across trials, I mean, sessions, is really important.
Yeah, this isn't just a random phenomenon. We can decode the past, right? We know what happened. It could be useful for memory. Absolutely, we're sure of that.
Yeah. Next question is by Carly Sumbrick. Do you think that it is possible that changes in perception of timing are facilitated by tuning changes in these time cells?
Oh, yeah. That fits into the equations really good. So briefly, we can change from time cells to place cells by modulating by the velocity. Like the chain rule from high school calculus. D by dt equals t dx d by dt. So you can turn stuff, and I might have flipped that. So you can turn stuff by modulating by velocity. You can turn time into space, right? You can get the Laplace transform of position rather than time. And so the way we do that is by having the way that we would do that mathematically. What it means is you change essentially the rate constant of every neuron in the population together by the same amount by dx dt, and that's understandable as changing the gain on the FI curve of these neurons. So you can imagine something like arousal or some expected time of occurrence affecting the population in the same way. And by changing the gain on these cells, you'd make time go faster or slower, regardless of what the mechanism is that you use to get these slow time constants. If you modulate them all together, you change the rate of time in the network. And so, yeah, we've used that a lot in making place cells and border cells and splitter cells. And we can make a passable model of the diffusion model for evidence accumulation. You have instantaneous evidence about which alternative you're choosing from. We can build out stuff that looks kind of a lot like populations of evidence accumulator cells in LIP and elsewhere. So yeah, absolutely, positively, it can be done. The big question, it comes back to this question earlier about MEC. If I'm at this point kind of convinced that these ideas make sense of a ton of stuff, right? But there's nothing in the equations that tell you how to fit the parameters, how to choose what is the gain for a different population, or whether this is modulated by velocity or this is modulated by the rate of information accumulation or expectation, or whether these cells are responding to pictures of cats and dogs and these other cells are responding to odors, and how those are distributed or flexibly changing across trials. So the theory doesn't tell us anything about that. And I think we should, I think we as a group should we should take these sort of ideas and look through the lens of this this nascent theoretical framework of the data we have in hand and think about new experiments to map out the brain.
Yeah. Well, thank you, Mark. That was fantastic. I love hearing.
Thank you for having me. Thanks for the great questions.
Ideas for things that bring everything together and might actually work, which is awesome. I think this is one of those. Yeah.
Good.
All right.
Great to see you.
All right. We're gonna
in real life. Okay.
Yep. Bye, everybody. Thanks for coming.
It's gonna be the same for me as well.
Can you guys quickly run through and could everyone introduce themselves? Just so that I can get the pronunciation of your names roughly correct. I can't promise I'm not gonna butcher your names, but I will try. The order we're gonna go in is Baharan.
Yes, Baharan was Ranjbar.
Can you say your last name? Okay.
Exactly.
Okay. And I think, and then Siddharth, Suresh.
Yeah, Siddharth Suresh, or you can just call me Sid Suresh, whatever works if you're finding it hard to pronounce the first word.
I will try. Yeah. I usually appreciate when people try to pronounce at least my name. Usually,
but it sounded fine.
Okay. Okay, good. And our host is?
That's me, Yuchi.
Yuchi. Okay.
Yuchi. Yeah.
Good. All right. What time zone are you guys at?
Eastern.
East. Okay.
And
you're the same as me?
2 p.m. Yeah.
Yeah. Sid?
It's 4 here. I'm at Central.
Mm-hmm.
I'm in Iceland. It's 8 minutes to 9.
Okay. Good, good.
9
Nice.
p.m.
All right, good. So what I was planning, wow, we never actually had such an early start. Usually we have like 2 minutes to go through everything.
Yeah.
So,
Would it be okay if I share my screen once and check if it works?
yeah, let's do that.
Well, I'm going to turn on my screen for a second. I'm still here.
Let's give people a second to join the meeting. All right. Welcome everyone to the next Neuromatch session. It's 2 p.m. in sunny California, but I suspect that it's all different times for you guys. I think by now you're probably familiar with how things work. Your questions ideally would go into the Q&A session. I will be monitoring that and I will read out your questions, time permitting. Otherwise, you can keep discussions going in the chat. So our first speaker today is going to be Bahareh Jozranjbar, who's a doctoral student in psychology at the University of Iceland in the laboratory of Dr. Heida Maria Sigurdardottir. Icelandic names, times. So without further ado, Bahareh, please take it away.
Thank you. Hi, everybody. My name is Bahareh, and I'm going to talk about the inseparability of visual and configural visual processing in dyslexia. Dyslexia is a problem in reading that occurs despite normal intellectual capacity, intact sensory abilities, and adequate educational opportunities. Dyslexia is often described as a phonological deficit, which means difficulty in manipulating and distinguishing phonemes that are the smallest units of speech sound. High-level ventral esteem regions in or close to the left fusiform gyrus that are important for recognizing objects tend to be underactive in dyslexic readers. This is consistent with some studies showing that dyslexic readers might have problems in object and face recognition. This is debated and not all the studies found that. In this study, we investigated whether dyslexic readers have problems with specific stimulus categories. If dyslexic readers' problems are visual, they could be restricted to visual word processing, or they might generalize to other visual categories, all or only some, as face recognition is assumed to require domain-specific mechanism, so faces might be distinct from other visual categories. We were also interested to see whether dyslexic readers have problems with feature or configural processing that are two different approaches to visual recognition. Feature processing is generally assumed to be important for word recognition, while configural processing is often considered the hallmark of face processing. Visual features of word is something like letters in the word. Just by changing one feature, we can change the identity of word. For example, here, by changing one feature, which is C, we change the identity of word, and now it's bat. For changing the configural processing, we change the distance between the features of object. But this does not necessarily change the identity of word, as you can see in the cat word. It seems that configural information is less likely to influence reading ability, but there is evidence showing that experts use greater holistic or configural processing to read. In our task, we manipulated feature and configural information of faces and houses to answer our questions. For example, here we changed the feature information of faces. You can see that eyes, nose, and lips are different between these two faces. Here we did the same for houses. Windows and doors are different between these two houses. Here we change the configural information of faces. As you can see, features are the same, but the distance between them are different. For example, we change the distance between the eyes, nose, and lips between these two images. And we did the same for houses and changed the distance between windows and doors, while features were the same. In our task, we first show them a fixation caress for 500 milliseconds, which was followed by a sample that could be a face or house for 300 milliseconds. Then there was a visual mask for 200 milliseconds, and then we showed them two images, where one of them was identical with the sample and the another one was different, featurally or configurally. The images remained on a screen until the participant indicated which one was identical with the sample. Comparing the overall accuracy of both groups revealed that there was a lot of variability, but on average, dyslexic readers were worse at recognizing houses, but probably they had no problem with faces. However, it seems that it doesn't matter whether it was featural or configural processing. We did not expect this result. What overall accuracy misses is that by relying solely on overall accuracy, the relation between different conditions will be overlooked. Here, for example, we have two conditions, A and B. The darker color means that they are more correlated. If we only compare the overall accuracy of A between two groups, we will miss the relations between A and B. Two groups might be equally good at condition A, but use different strategies for doing that. If A and B tap into different skills or require different strategies, accuracy of a trial of task A should be more correlated with the accuracy of another trial of task A than a trial of task B. Then the left pattern will be our conceptual model that assumes A and B are independent skills or conditions. If A and B are not independent, but instead tap into the same skills, then a trial from A should be no more correlated with another trial of A than it is with a trial of B. And we would see the right pattern. How will this pattern correspond to the real data is what we are looking at in the representational similarity analysis, where we compare the conceptual models to reference models. Reference models are the correlation between feature and configural processing of faces and houses for dyslexic readers and typical readers. Darker color means that they are more positively correlated. Here, for example, there is a low correlation between configural face and configural house. And here there is a high correlation between the featural face and configural face. These are the conceptual models for stimuli, process, and difficulty level. If these patterns correspond to reference models, that means that they are independent skills. Here, for example, is the conceptual model for a stimulus. These two are both faces, so they are highly correlated. And here, one of them is face and another one is a house, and they are not correlated. It is the same for the conceptual model of the process. These are both featural, so they are correlated. And here, one of them is featural and another one is configural, so they are not correlated. And it is the same for difficulty level. What we found was that for typical readers, the stimulus conceptual models corresponds to the real data. Some typical readers seem to be good at face recognition and some at house recognition, but they are not necessarily the same people. This shows that face and house recognition are two separate skills for typical readers. We found that pattern of the conceptual model of process in the real data of typical readers, which means that they act differently based on different processes. We did not find the pattern of conceptual model of difficulty level in the real data of typical readers, and we did not expect to find this pattern because if a participant performed well on easy trials, then he or she might also perform comparatively well on more difficult trials. We also found a stimulus pattern for dyslexic readers. However, we did not find any indication that dyslexic readers act differently based on different processes. We also did not find the difficulty level pattern in dyslexic reader data as we expected. The representational similarity analysis showed that typical readers acted differently, both depending on whether the stimuli were faces or houses, and whether feature or configural processing was task relevant. Dyslexic readers performed differently based on a stimulus category, but not process. It seems that dyslexic readers solely relied on one process when analyzing the stimuli. It is possible that if dyslexic reader cannot flexibly change the way that they process a stimuli depending on what is relevant, it affects their reading. As to read efficiently, both featural and configural processing are required. I would like to thank my supervisor, Heida Maria Sigurdardotter, and Arne Christiansson, and my collaborator. And thanks you for listening. Thank you.
Thank you. I think we will have to stick with sort of virtual applauses at the end of these presentations. So we have a question for you in the Q&A. Have you considered doing a similar feature role versus configure role contrast in a word recognition task? What would you predict there in accuracy and RSA?
We did not do that. We didn't do that, but we are going to do it. It's an interesting possibility, and I think that it should be the same, and they shouldn't be able to discriminate between feature and configurable processing, as they weren't unable to do that for both faces and houses, and it shouldn't be that different for word recognition, I assume.
Thank you. So I had a question that may sound a little bit silly, but so a basic concept in architecture is that they sort of design houses to be a little bit face-like. You know, they have two windows and a door in the middle that kind of symbolizes a like a nose, and sort of the whole shape, you know, in a very abstract way, kind of resembles a face. And I'm kind of wondering whether this could potentially confound your research a little bit, that you know, at some very abstract level, faces and houses are quite similar, or is this similarity actually intended in your task?
We actually did that for, we had the house recognition for control, and we chose that because it was some sort of like the face, and we could change the feature and configure information of houses just like we did for the faces, but it wasn't easy for other stimuli. But in our future study, we are going to do the same analysis on different type of the objects to see what will happen.
All right, great, thank you very much. So if there aren't any more questions, then we will move on to our next speaker.
Thank you.
Thank you very much. Our next speaker is going to be Tyler Bonan, who is a PhD student in Dr. Anthony Wagner's laboratory at Stanford University. Tyler, feel free to take it away.
Was that good?
Yeah.
Great. So yes, I'm a graduate student working with Anthony Wagner and Dan Yeamans. They're dual advised. I want to just begin by saying, wow, this is a really beautiful forum. That's a really important step, I think, towards democratizing a lot of our scientific practices, mitigating our impact on the environment, and just like having beautiful conversations and doing better science. So I'm super happy to be here. Super happy to be talking about the work that I've been doing with Anthony Wagner and Dan Yeamans. This project is really our first step towards building biologically plausible computational models of mammalian memory systems. Specifically, we're looking at the medial temporal lobe, which is a network of neural structures that have been conserved across hundreds of millions of years of evolution. These structures are typically implicated in memory-related behaviors. For example, the medial temporal lobe supports a lot of like one-shot learning behaviors like episodic memory, familiarity-based recognition, etc. Yet, there's an enduring debate surrounding the involvement of the medial temporal lobe in perception. Within the object perception literature, this debate centers on the role of perirhinal cortex, which is a medial temporal lobe structure which is in the primates situated at the apex of the ventral visual stream, and is often considered to be a gateway between visual and mnemonic information processing. I'm going to show you just a lateral view to situate you about the neuroanatomical relationship between the ventral visual stream and perirhinal cortex. Just showing you that one of the main outputs of like high-level visual cortex, like IT, is going to be perirhinal cortex. Now, over many years, there's been this observation or claim rather, that perironal cortex enables complex perceptual behaviors. That is to say, perceptual behaviors that would not have been supported by the ventral visual stream alone. And in order to cash this idea out in an experimental context, what is typically done is the administration of concurrent visual discrimination tasks, so-called oddity tasks, to perironal lesioned and intact subjects. And so control stimuli are designed to rely on canonical sensory regions and be unaffected by, rely on canonical sensory regions and be unaffected by perironal lesions. For example, this size discrimination task, where the oddity here is the bottom left. Color discrimination task, again, oddity is the bottom left. And the object discrimination task, again, the oddity is the bottom left. And these are all tasks that are thought to rely on canonical sensory cortex. Conversely, diagnostic stimuli are thought to, or designed to require so-called complex perceptual representations. And these are what should be used to evaluate perironal cortex's role in perceptual processing. And I'm just going to show you a couple of examples of different kinds of operationalizations of this idea of stimulus complexity. Now, for about 30 years worth of experimentation using perironal lesioned and intact subjects and administering these kinds of experiments to them, there's a rich body of evidence which suggests that perironal cortex is involved in perception. And there's also a rich body of evidence which suggests that perironal cortex is not involved in perception. What I'm hoping to do today is just to share some data with you which suggests that situating human performance in relationship to a linear readout of the ventral visual stream will enable us to disentangle these seemingly inconsistent experimental outcomes. That is to say, there's actually no inconsistency here. What's inconsistent are just the descriptions that we use to describe these data. And so let's just kind of like flesh out this idea a little bit. If we have ventral visual system supported performance here on the x-axis, going from chance to ceiling, and on the y-axis, we have human performance going from chance again to 1. The diagonal would just be the pattern of behavior predicted by a linear readout of the ventral visual stream, where the hypothesis is that perironal cortex enables neurotypical human subjects to outperform IT, outperform the ventral visual stream. And in the absence of perironal cortex, perironal lesion subject behavior should revert to what you see or what you would expect from the ventral visual stream. Critically here, if the ventral visual stream was at ceiling and humans were below ceiling, this just suggests that while the ventral visual stream is sufficient to do this task, there's some downstream processing that is required. And so these stimuli would be not diagnostic of the particular hypothesis that we're trying to evaluate here. And of course, the big question is just, well, how do you determine what is ventral visual system supported performance, right? And what we're going to do today is use convolutional neural networks as a computational proxy for the ventral visual stream. A couple of steps here in order to kind of just convince you that we can do that. We're going to take a bunch of stimuli that were presented to 2 primates in a study by Majaj et al. 2015. We're going to take those same stimuli where electrophysiological data were recorded from those macaques. We're going to present those same stimuli to a model. We're going to extract from all layers of that model. We're going to extract responses, and we're going to try to learn to predict the neural data in a cross-validated way. And so we're going to do that both for neural area V4, and we're going to do that for neural area IT. And what you can see in the plot here is that earlier stages of the model better predict V4, later stages better predict IT. And so we're going to take this model and we're going to estimate model performance from an IT-like layer. In our oddity task, we're going to take this choice array of n objects, segment them into n independent images. We're going to present these images to the model, extract n independent feature vectors, and compute the pairwise correlation between them, generating covariance matrix. And we're just going to determine the item with the lowest mean off diagonal correlation. That item is going to be identified as the oddity here. And we're just going to label that trial as either correct or incorrect, depending on whether or not the model got ground truth. Great. So we're going to take this model, this computational proxy for the ventral visual stream, and we're going to use it in a retrospective analysis. That is to say, we're going to collect a bunch of behavior and stimuli from relevant published experiments administered to perironal lesioned and intact subjects. We're going to estimate model performance on all these experiments. And we're going to compare model performance with perironal lesion and intact behavior. I'm just going to cache this out first in a set of 4 experiments within one study from Behrens et al. Experiment 1, high ambiguity novel objects. We have perironal lesion subject behavior on the y-axis and model performance on the x. Second experiment, high ambiguity familiar objects. And what I'm doing here is showing you the averaged performance across all trials. Low ambiguity novel objects and low ambiguity familiar objects. We can do this, of course, not only for these 4 experiments within this one study, we can do this also for all of the experiments that we have access to. And what you can see here is that perironal lesion behavior seems to be predicted by this computational proxy for the ventral visual stream. That is, of course, evidence that the model is a decent model, but it's not yet evidence that perironal cortex is involved in perception. To do that, to convince you that, I would have to show you that perironal intact behavior is off diagonal, is significantly outperforming this model for the ventral visual stream. So again, I'm just going to show you the perironal lesion behavior here, kind of abstracting away from the details of which study it was. Now, perironal intact behavior is significantly outperforming this computational proxy for the ventral visual stream, such that there's a significant interaction between the slope of these 2 lines. We can say here that perironal intact behavior diverges both from the ventral visual system model and perironal intact behavior. And we can kind of aggregate all these data into one plot and show you not only the experiments that we think are appropriate, but we can also show you that there's a lot of experiments that fall on this x=1 line, right? That is to say, there were a bunch of experiments in the literature that our model would suggest have been misclassified and should be discarded. And you can see that very clearly. There are these 3 distinct distributions, right, in the model performance space. Critically, if we look at the behavior of hippocampal lesion subjects, it doesn't seem like there's any difference between hippocampal lesion subjects and hippocampal intact controls. So it appears that perironal cortex within the medial temporal lobe is what's supporting these behaviors. Now, you might be wondering, hey, could these deficits be due not to damage in perironal cortex itself, but perironal adjacent sensory cortex, like, for example, IT? And so I'll just remind you that we have a model which has like differential correspondence to early and late stage process of the ventral visual stream. And instead of just showing you model performance on this retrospective data set for the IT-like layer, I can show you model performance on the retrospective data set for a whole bunch of layers, basically all of them, right? I'm just gonna visualize 4 of them here. And what you can see is on each of these columns here is a layer of the model. And lower row here is hippocampal lesion subjects in their control. Top row here is perironal lesion subjects in their control. And what you can see is, I'm just visualizing to show you that across all the layers of this model, there doesn't seem to be that big of differences between each of these layers. And so what I can show you is that I can take this model, this measurement that we have in the plot on the left, this delta IT minus V4 estimate, that is, what is the differential prediction of IT responses over V4? And I can compare that to a differential predictivity for perironal lesion subject behavior. And what you can see is in red/pink/purple, what you can see here is that the better the model fits IT, the better that model layer predicts perironal lesion subject performance. And this observation is not held in the hippocampal lesion subject data. And so to answer this question at the top of the slide, no, it doesn't seem like we could explain what's going on in these data by just simply suggesting that there's damage in adjacent IT cortex. But as you can see, as you kind of sweep across these slides, it doesn't seem like these stimuli allow you to really disentangle V4 from IT supported performance, right? That is to say, IT-like model layers look a lot like V4-like model layers, which is not really ideal for our purposes. So there are a bunch of other architectures. I'm just gonna maybe take like one more minute to finish this up. There's a bunch more architectures that we could perform this analysis with and show that across all the state-of-the-art model architectures, none of these model architectures are predicting periodontal intact subjects any better than the initial architecture that I showed you. And we can also validate these initial findings with novel experiment where we're looking at electrophysiological recordings in IT and periodontal intact subject behaviors. And what we effectively want to do is take these new stimulus sets, and we want to compare model performance on this new stimulus set with electrophysiological recordings in this new stimulus set and periodontal intact performance on this new stimulus set. And what you can see here is that IT is clearly separable from V4 behaviors. And the farthest left panel, the model is able to predict the IT supported performance, but periodontal intact subjects are significantly outperforming the model here and IT. Interestingly, we can see that periodontal intact divergence from IT scales with reaction time on the far right. So conclusions here. The medial temporal lobe is necessary for neurotypical performance on concurrent visual discrimination tasks. Periodontal intact humans outperform the ventral visual stream and deep learning models of vision. Our future work is going to be guided by twin imperatives, right? The stimulus computable models will help us better understand the function of the medial temporal lobe, and biological constraints might enable us to better develop models that achieve more animal-like behaviors. Thank you to both labs. Thank you to all the original authors and to a bunch of people who helped me along the way.
Thank you, Tyler. So while we wait for potential questions from the audience, I was wondering how well would the computational model that you guys came up with generalize? So, you know, IT has specific roles in visual discrimination. Empty area would be, for example, more, you know, movement type discriminations. I think there are a lot of classical experiments on that on primates. Would your architecture generalize to something like that? Or can you take it even further? Would it generalize to auditory type discrimination, or even further some more complex cognitive functions? Or do you think that the architecture that you're looking at here has to be very specifically used for this particular task?
Yeah, so I would imagine, I think it's a fantastic question. I really love that question. I would imagine that we would generalize well to, for example, auditory discrimination tasks. I would imagine you would see a similar pattern of performance where for certain kinds of auditory discrimination, you might need something like periodontal cortex and the kind of strictly kind of feed-forward architecture that you see not only in the ventral visual stream, but also throughout auditory cortex. I would imagine that these results would hold in auditory cortex. I'm not sure if they would hold in something like motor cortex, not sure. There's been some work that's suggestive in that space, but I wouldn't be able to tell you if you can formalize it like a feed-forward process. My model might help you understand that process, but if you can't formalize it like a feed-forward process, which is often what you need in motor tasks, yeah. But I think it's a fantastic question.
Fair enough. Thank you. So there is a question in the Q&A for you. Fantastic talk, blurred through the last slides, but I assume I can reach out to you via email for those slides. So yeah, if there's something like this coming up, you know, you can probably, if you're happy to share your email address, you can type it into the Q&A and sort of take it from there. Yeah, if you guys have any more questions to the speaker, feel free to type them into the Q&A. I think he will try to answer them there. And we are going to, in the interest of time, we are going to move on to our third speaker in this session, Siddharth Suresh, who is a research assistant in Dr. Emily Ward's laboratory at University of Wisconsin, Madison. Sid, would you like to take it away?
Yeah. Hello, guys. I'm Siddharth Suresh, and I work as a research assistant advised by Dr. Emily Ward at the Visual Cognition Lab in the University of Wisconsin-Madison. This is my very first academic talk, and I'm excited to present my project to all of you. In this talk, I'm going to talk about the emergence of visual ensemble representations in deep neural networks. With Halloween coming up, I'm really excited to start seeing candies all around. Besides from knowing that these are Halloween candies, these are very interesting visual stimuli for a couple of reasons. Just by looking at these images, there's a few types of visual information that we can recognize. We can perceive what the average shape is. The starbursts on the left have a square shape, and the assorted mixture of candies on the right are roughly bean-shaped. We can also recognize the average color of these images. The average color is not explicitly present in the images, but we have the ability to compute it. Surprisingly, we can also recognize the color diversity, that is, if the colors come from a diverse set or not. All of these properties, the average shape, the average color, and the color diversity are examples of different types of ensemble perception. Ensemble perception refers to the visual system's ability to extract summary statistical information from groups of similar objects, often in a brief glance. Today, I'm going to be focusing on color diversity. It has been found that people perceive color diversity without actively paying attention and without perceiving the individual elements that make up the image. All of what I've told you suggests that humans automatically and efficiently extract ensemble properties. Such ensembles are thought to be a solution to the capacity limitations of the human visual system. At the surface level, this has a lot of similarity with object recognition, which is something we also seem to do somewhat automatically and very efficiently. But object recognition differs from ensemble perception in a couple of different ways. In a lot of cases, colors are often arbitrary and not very meaningful while recognizing objects. Hence, when it comes to object recognition, we may not want to retain the color information. It may be helpful to generalize over color and use other features instead. The primary question that I am going to be answering today is, do ensemble representations arise in a visual system that only recognizes objects? Given that colors can be arbitrary, they may not be encoded in representations when you're only doing the task of recognizing objects. Apart from object recognition, humans do a lot of different tasks, but we now have artificial visual systems that are good at only recognizing objects. And I'm going to be using these systems for our experiment. Deep neural networks are known to be modeled after primate vision. The information in deep neural networks is coded hierarchically as we move from shallower layers to deeper layers. Shallow layers and deep layers are the layers that are closer to the input and the output respectively. The representations within these layers become more sophisticated as we go deeper into the network. We can extract these intermediate layer representations, which make deep neural networks good tools for studying the visual representations underlying various perceptual tasks in a visual system. These representations comprise of different number of units depending on the layer which they correspond to. In our experiments, we used a ResNet-50 deep neural network, which was trained to recognize objects. This network takes an image as its input and predicts the objects that are present in the input image at the output. The network is trained using 14 million images to recognize objects belonging to 1,000 different classes. We refer to this as a pre-trained network. In the slide, an image of a boat is presented to the deep neural network, and the network extracts features from the image. The features turn into more sophisticated representations as they move deeper into the network. The network uses the representations from the pre-final layer to identify the presence of the boat. Kindly note that the illustration in the slide is used to explain how the deep neural network in our experiment works and does not accurately depict the architecture of ResNet-50. Now that we have the deep neural network that we will be using, I'm going to answer two questions. We'll first find out if color is represented in these networks, and then we'll see if the ensemble property of color diversity is represented in the network. First, I will be focusing on color representations. To answer this question, we choose images that represent everyday objects. These images were designed to make sure that the objects could be of any color and were not associated to a specific color. We use a stimulus set containing 540 unique objects, each being red, green, and blue in color. These stimuli were presented as an input to our pre-trained neural network, and we extracted intermediate layer representations starting from the shallow layers. Each of these intermediate layer representations comprises of neural-like units, which were used as an input to a linear classifier. The linear classifier was trained to identify the color of the object present in the image. This process was repeated for the deeper layers. The linear classifier was trained on different number of randomly sampled units for all the intermediate layer representations used in our experiment. We varied the number of randomly sampled units to see how the color information was distributed among the units in a layer. We also wanted to see how the performance varied as we increased the number of randomly sampled units. We randomly sampled 500, 200, 100, 50, 20, and 10 units in our experiment. The x-axis in the graph refers to the names of the different intermediate layers used in our experiment. As we move from left to right on the x-axis, the layers become deeper. This means that conv 3 is farthest from the output, followed by conv 2, and the pre-final layer is the closest to the output. The chance level accuracy of this task was around 33%, and the classifier was able to perform better than chance regardless of the layer and the number of units. All the points are statistically above chance except for the most sparsely sampled point in the pre-final layer. All the slopes are also significant with a p-value of less than 0.01. Hence, we can say that color information is present in shallow, intermediate, and deep layers of the network. It decreases in deeper layers and reduces with more sparse representation. What about object categories? Are they still distinct? To assess this, we used t-SNE to visualize how layer representation vectors cluster. t-SNE is a dimensionality reduction technique that is often used to visualize high-dimensional vectors in 2 dimensions. If color information is dominant, we would expect to see clusters that are roughly organized based on color. And if the object information is dominant, we would expect to see tiny clusters, each corresponding to object categories. In the earliest layer, we see a little bit of object clustering, but primarily the organization is by color. In the next layer, the organization of the clusters changes to being organized much more by objects. And in the pre-final layer, clusters are very clearly organized by object category. What this shows is that object categories are still being preserved, which is, after all, the goal of the network. It also shows that color information, though classifiable, gets more suppressed compared to object information in deeper layers. So, firstly, I pose the question of if the color is represented in the network, and we see that color is represented at all layers of the network. We also see that object categories are preserved. Now we go on to see if ensemble properties are represented in the network. I started off by introducing you to this idea of color diversity using Halloween candy. Clearly, we are not using these images for the experiment. The type of stimuli that I'm going to use is a little simpler, and they look like this. This is a simple way of presenting color diversity that has previously been used on human subjects. The colors of the letters in the stimuli were sampled from the color wheels shown, depending on its diversity. For low diversity images, four colors were sampled from 6 adjacent colors on the color wheel. And for high diversity images, 4 colors were sampled from the 19 colors present on the color wheel. The stimulus set consisted of 12,600 images, half of which were low diversity images and the rest for high diversity images. We used the same approach as earlier, and since we are still interested in knowing how the color diversity information is distributed within units, we use the same sampling approach as well. The chance level accuracy of this task was 50%, and the classifier was able to perform way better than chance regardless of the number of units and the layers again. All the points were statistically significant, and all the slopes were also significant with a p-value of less than 0.01. Hence, we can say that color diversity is present in shallow, intermediate, and deep layers of the network. We can also say that it decreases in deeper layers and that it reduces with more sparse representation. We use this type of stimuli since it has been used before with people, but low and high is a pretty rough measure of color diversity. In all these cases, we can actually get a measure of color diversity by calculating the average variance of each image. The average variance is calculated as the average of the variance of all the non-zero pixels present in each of the color channels that make up the image. Getting a person to tell you these numbers is pretty difficult, but getting a network to predict these continuous values is a lot easier. And we'll use regression now to see how well the regression fits the continuous value of color diversity. Here, the only thing that we changed is from earlier is that we now use linear regression to predict the continuous value of average variance. What's now important is to see how these networks model variance. I would now be plotting the R-squared. We find that the 500 randomly sampled units account for about 75% of the average variance in color diversity. The R-squared value increases as we increase the number of sample units. There is no clear trend about how the average variance fit changes across the layers for the same number of sample units. So are color ensemble properties represented in the network? Yes, color ensembles are represented in all layers of the network, and they account for quite a bit of variance while predicting color diversity. I would like to conclude by saying that deep neural networks trained for just object recognition are sensitive to color diversity, indicating the presence of ensemble representations. Even though color may not be meaningful to recognize an object, deep neural networks retain the information of color and the relationship between these colors. Unlike humans, deep neural networks have no computational capacity constraints, and yet we find the presence of ensemble properties in them. Deep neural networks can be useful in studying the neural mechanisms of ensemble perception. Thank you guys for listening to this talk, and I would like to thank my advisor, Dr. Emily Ward.
Thank you, Sid. Awesome talk. We already have a couple of questions for you. So I am going to try to go in order of popularity. The first one being, is there a databasis here? Since although there is 50% low and high diversity image split, the combinatorial complexity of the high diversity images should be much higher, requiring more training images.
I'm sorry, I lost you in the middle there. Could you please?
Do you have can you open your the Q&A?
Yes, I just opened
at the
it.
bottom. Right, so this is the.
Is there a data? Okay.
Right. It's the question from Cole Hurwitz. Is there a data bias here? Since although there is a 50% low and high diversity image split, the combinatorial complexity of the high diversity images should be much higher, and hence requiring more training images.
This is a very good question. So firstly, I'd like to say that the network that we used was pre-trained on ImageNet, and it wasn't used like we didn't use these and our images, our stimuli to train this network. And secondly, we based this off of a paper by Bronfman where they and Dr. Ward, who evaluate the same stimuli on humans. But maybe we will go back and look at this further.
Thank you. And I think we have time for one more question. You mentioned that no clear pattern emerged for color diversity across layers, but for nearly all samples, it looked better at cone 4. Perhaps this is showing an intermediate level optimization. This is the top question right now.
Yes, but we did see that, I mean, the trend was visible, but then for the least number for like sparse sampling, we don't see this trend, which is why since the project is still in works, we'd like to go investigate this in more detail.
Great. I think we have time for maybe one more question. I think this is a short one. Have you tested this neural network's performance on black and white images?
I have not tested the neural network's performance, but I'm pretty sure there's a lot of literature which checks how these how Resonance 50 performs, Resonance 50 which is pre-trained on ImageNet performs on black and white images.
All right, great. Thank you very much. I think we will stop here in the interest of time, so that we don't get kicked off the Zoom room. I would like to say thank you to all three of our panelists. I think you guys did a fantastic job today. And I would like to say thank you to Yuji, our backend, who managed all the technical parts of this session. So thank you again all, and have a fantastic rest of your day, rest of your afternoon.
Thanks a lot for hosting this.
Sure thing. Have a great afternoon, guys.
Are pretty varied. I think they're going to be pretty good. So we can, let's see, it's 3 o'clock right now. We can go ahead and get set up with our first speaker. So Michael, why don't you turn on your camera and share your screen here? And we will have our first speaker is Michael Cohen from the University of Pennsylvania. And the title of his talk is The Effects of Memory for Emotional Valence and Episodic Detail on Decision-Making in Aging. So why don't you just go ahead and take it away, Michael, whenever you are ready.
All right, and can you guys see my screen okay?
Yep, looks good. Here we go.
All right, great. Okay, so what I'm going to be talking about is how people can use memory of past experiences to make good decisions. So you can think of where that might be relevant in contexts like investing, dating, voting. Those are all cases where like your past experience with an individual or with a class of or some type of choice, like that should guide your future decisions. So in cases where you have repeated related experiences, so in those cases, reinforcement learning could influence choices. But if you only have one prior experience, then you might have to rely on explicit memory. So a key question that we were asking with this study is whether age-related impairments to explicit memory could lead to impaired decision-making in these kinds of situations. And then a related question is more about the mechanism. So whether these decisions rely on general memory for valence, so like just like something is good or bad, or whether they rely on memory for specific episodic details. And so this question is also important in the context of aging because aging tends to impair memory for specific details, but memory, general valence memory tends to be preserved. So we used a task that was adapted from Deepu Murthy and his colleagues. And so we showed both social stimuli and non-social stimuli, although actually the data I'm going to present today, we're combining across both of those categories. So the social stimuli were in the form of a dictator game. So basically, you're told that you have another player who the face represents. They decided to share how they were going to share $10. And they could either give you $5, which is the high reward condition, or $0, which would be the low reward condition. Or there's a non-social condition where it's framed as a lottery, and this house is associated with getting either $0 or $5. A 5-minute distractor task after that. And then you get the decision task. And this is kind of our key behavioral outcome. And the choice is whether you're going to play again with the pictured face or house, or whether you'd rather take your chances with a randomly chosen new face or house. And so the hypothesis is that what you remember about the previously seen face or house, that should impact the decisions that you're going to make. And then finally, we gave a memory test. So first there was a test for item memory, just do you remember this face or house? And then associative memory, so do you remember how much they gave you? And then finally, a confidence judgment. So we tested older adults from the Penn Aging Brain Cohort, and this is a group that's been pretty carefully screened by the neurologists at the Penn Memory Center, so they're all defined as cognitively normal. Some of these people were run in the MRI scanner, some of them were only run behaviorally, and we have a middle-aged sample. So at this point, we only have behavioral data for middle-aged adults, although we are going to start scanning middle-aged adults as well to have a comparison of the brain data. And then data collection is ongoing. It was paused for a while with the pandemic, but we actually have recently resumed. So assuming that the third wave doesn't hit Philadelphia too hard, we're going to keep collecting data. So hopefully there will be more solid data soon. Okay, so so item memory, generally we see that middle-aged adults are doing better than older adults, but older adults are still well above chance. Associative memory, again, middle-aged adults are doing a little bit better. Older adults are only barely above chance there. But then the key thing is if we limit it to confident associative memories, there we see that both middle-aged and older adults are pretty solidly above chance, although really not great. I mean, the middle-aged adults, even when they're confident about their memory, they're still only a little above 60%. So it's still not great, but there is some actual memory there. And then again, the older adults also are a little lower, but still above chance when they're confident. So then the kind of essential behavioral question is whether associative memory is needed to make adaptive decisions. And what we see first is when people say that they're guessing about their associative memory, their choices are basically random. So the middle-aged adults may be slightly above chance in their choices, but generally they don't seem to have much basis to make their choice when they don't remember the value that the person or house gave them. When their associative memory is confident correct, there we see that people are making adaptive choices in both age groups. So basically that means that if you're confident and correct about how much you got from that stimulus, you will choose to play again a lot more often with the high reward stimulus than with the low reward stimulus. Although you can also see that there is notably weaker preference on the confident correct items for the older adults versus the middle-aged adults. And then finally, when they're confident incorrect, choices are reversed, which also makes sense because if it was actually $0 and you think it was $5, or vice versa, you'll make the completely wrong choice. But again, that goes with this idea that people are relying on memory when they make their choices. So then the next question is, what are the neural correlates of memory when older adults are making decisions? And so again, we had fMRI data for some of the subjects. And what we're looking at here is contrasting adaptive decisions versus maladaptive decisions. So adaptive means you chose to play again with a high value stimulus or you chose to avoid a low value stimulus. Maladaptive is the reverse, choosing to play again with low value or avoiding high value. And then we're only looking at trials where they did remember the item, combining faces and houses. And again, this is during the decision-making test. And so what we see is that there is some evidence that general valence memory is involved. So we see the amygdala is more active on adaptive choices. But then we also see that the medial temporal lobe is also more active on adaptive choices. And that's suggestive of specific episodic detail memory. Now, again, this is still pretty preliminary. We're not cluster correcting. It's only 11 subjects so far, but we are getting more data on this. But the conclusions from this part are that older adults can make adaptive decisions based on explicit memory, but there are some subtle impairments in both memory and decision-making. But the preliminary MRI data suggested that both emotional valence memory and specific detail memory was contributing to decisions. So we did a second study just to separate these two mechanisms more behaviorally. And so in study one, the values were always either $0 or $5. So the key difference in study two is that we used a wider range of values within the high value and low value categories. And so that lets us separate out memory for episodic details from memory for valence. And so we did here, so we had, again, the study reward phase, but again, so they got the offer, which could vary, and I'll explain in a minute exactly how it could vary, but those are more variable. Then they got the item memory test. And if they said they remembered it, now they're getting a multiple choice test for associative memory. And of the four options, they have the exact amount, and then they also have a lure. And the lure is the same valence as the correct amount, but not actually the right value. They're also judging their confidence, whether they're confident in the specific amount within $1, just guessing. And we're defining specific memories as being correct and confident in the exact amount, because we're assuming that that should be like a really precise episodic memory, and so you should have that level of confidence. To give a few more details, again, it was 50% low reward, 50% high reward. The stimuli could vary over the course of $1.50 in 10-cent increments. The lure values were always within $1, the exact value. Participants were recruited using MTIP. This was earlier on in the pandemic. We tried to get a range of ages, But all the data I'm going to show here, we actually combined across age groups because in short, collecting aging data on mTurk is kind of weird and turns out to not be a great idea. So we're still wanting to get at that a little bit better. But yeah, so the data looked like it made more sense when we just collapsed across age group. And okay, so first the memory results. So we see that memory was above chance for the exact values. So there was some degree of precise detail memory. Also, even when they didn't have the exact memory, lure memory was also above chance. So it's a little bit better for low value than high value, but regardless of the reward value, they are having some memory for general valence, even when they don't get the exact value correct. And then also looking at confidence, so they had better memory, or sorry, yeah, when they were confident in the exact amount, they had a fair amount better memory in the exact value than when they were just confident about the valence. And you would expect there to be confidence again, because like I had said, we assumed before, if you're correct on the exact amount, that probably means that you have a really detailed episodic memory. And then valence, also there was some effective confidence, although it was not quite as strong, mostly in high value items. But if you're confident in the valence, you are at least a little bit more likely to be right about the valence. Okay, so first we see the decisions, basically we do replicate the effects from experiment 1 when you collapse across exact values and lures. So when you knew that it was a high reward item, you're more likely to choose it, basically. And if you were confidently incorrect, then you show a reverse pattern. Again, that's what you would expect. But the new thing with this experiment is that we were able to divide up the confident correct items based on whether they only had the valence confident incorrect or whether they also had the exact value confident incorrect. So what we see is that when they only had the valence confident incorrect, there is still a pretty strong effect of valence on their choices. So that suggests that that valence is sufficient to lead to a pretty strong effect on choice. But we also see that when they actually do have the exact values, then that's actually an even stronger effect of memory on their choices. So that's suggesting that there is some additional component of the exact value influencing their choices. So the overall conclusions are that it seems like people in all age groups can use explicit memory to make decisions. Memory for emotional valence, which we think is mediated by the amygdala, that probably is a key mechanism. It is worth noting that it's kind of interesting that these memories are being formed on a single learning trial, even when the emotions involved aren't really that strong, but it's still enough to get the amygdala involved. But it's also important that when memory for a specific detail is available, it leads to even more robust performance on the decision-making task. And then finally, looking at the aging piece, so older adults are showing mild impairments on the decision-making task, and that especially shows up as a weaker preference for adaptive choices when they're confident correct in their memories. But overall, the pattern of behavior is consistent with prior findings from older adult memory, which is that you get spared memory for valence, and we think that's potentially underlying a lot of the successful decisions, but then impaired memory for specific details would lead to these subtle impairments in the decision-making. So I want to thank my collaborators on this, Carolina Lempert, Joe Cable, and Dave Lote. So the three of them actually initiated this project, and I kind of jumped in a little bit later, and then as well as the RAs on this, our funding sources, and all of you for listening.
Fantastic. Thank you, Michael. And because we cannot applaud, if you enjoyed the talk, please say yay or thank you or something in the chat. And make sure you set it to panelists and attendees so we can all see it. If you have any questions for Michael, we can go to the Q&A. I didn't see any pop up during the talk, but that doesn't mean that you can't use that box now. And while we're waiting for questions to roll in here, Michael, I've got a question for you. So we talked about something kind of similar to this back whenever I visited Penn in like March or February before everything went crazy. And I remember us talking a little bit about metacognitive sensitivity. And I was wondering whether you'd looked into or had any plans to look into either the effect size of the difference between the confidence levels or correct, incorrect across older versus middle-aged adults, or whether you were thinking about doing any metacognitive sensitivity measures.
So we weren't necessarily on this data set. Actually, I mean, the conversation we'd had was actually a different data set that I still haven't.
Okay.
But I'm interested in metacognition, and yeah, I mean, any way to get that stuff in, I think, is still cool. So yeah, I mean, I'd have to think about whether it's relevant, but it does. Yeah, I mean, it seems like our data probably would kind of that, but we haven't looked at it yet.
Cool. Yeah, I think it would be fun to look at that and how it might interact with some of the neural measures that you're looking at, like how the amygdala reactivity or other measures that you have would be differentially predictive of metacognitive sensitivity, maybe in the middle-aged versus the older adults. So cool. Does anybody have any other questions for Michael? Okay, well, Michael, you can feel free to drop that website in the chat if you want to have follow-up discussions. And of course, these follow-up discussions are welcome for any of our speakers. So feel free to ask. We've got a quick question here, very quick, because we will need to go to the next speaker in just a minute. But a question from Parnian is, I was wondering if the task and codes related to that might be available on OSF somewhere.
So we haven't posted it there, but you're welcome to email me. I can definitely share more with you by email. And yeah, I think I'm going to post a link to my website. But yeah, generally email me. I'm happy to share more.
Great, and we can drop some links in the chat as well to make it easy for people. Okay, fantastic. So thank you, Michael, for the fabulous talk. And we will now move on to our next speaker, but feel free to continue engaging in the chat and other measures that we have. So let's move on now to Dana Glenn, who is coming to us from UC Riverside, around the corner from me. And she is going to talk about age and stimulus type influence the association between multivoxel threat representations and explicit memory during generalization. And so Dana, why don't you go ahead and share your screen and take it away for us.
All right, thank you. Let's see. Okay.
Looks good to me.
Cool.
All right, take it away.
All right, great. So as Megan mentioned, my name's Dana Glenn. I'm a graduate student at UC Riverside, and I'll be presenting some preliminary work looking at how age and stimulus type affects threat representation during threat generalization. So this is similar to some work that we've published on in the past. So if you'd like to check that out, Megan's going to drop that in the chat. Due to some time constraints, I can't dive into the methods in as much detail as I want to. And so some of that will be on that paper. And of course, you're also welcome to ask me any questions. So fear is a very important emotion. Although it's unpleasant, it is an adaptive mechanism that allows us to predict, react to, and adjust to threat in order to avoid both present and future danger. Humans and animals can anticipate potential danger by applying previous knowledge about threats to novel situations. So threat generalization is the ability to identify features of stimuli or situations that resemble those linked to a past threat, and it's essential to our ability to learn about threats. So one important feature of a threat stimulus might be whether or not it has social implications. Because humans have evolved to work within complex societies, it's important for success and possibly for survival that we can precisely remember who is a friend and who is a foe. And while non-social threats can also pose significant danger, they might be more easily categorized as threatening or non-threatening. So for example, if this woman screamed at you, it would probably be beneficial not only to remember her, but to be able to tell her apart from other similar looking women. On the other hand, if this bear attacked you and you somehow survived, it would probably be a good idea to just avoid all bears in the future. You can categorize them all as dangerous without making judgments about subtle differences in how they look. So the specificity with which you need to distinguish between threatening and ambiguous stimuli may influence how much you generalize knowledge about learned threat to other situations. And there's research to suggest that stimulus type does influence threat learning. So people display stronger conditioning to evolutionarily relevant threats, and this conditioning is more resistant to extinction. But few studies have directly compared threat generalization elicited by social versus non-social stimuli. So our first research question was to test whether threat representations are influenced by social context. And we examined fear ratings and neural activation in several brain regions implicated in threat learning and threat generalization in a group of both adolescents and adults. So we were also interested, how does this change across development? The ability to differentiate between threatening and ambiguous stimuli increases with age differences persisting into adolescence. And so there's research to suggest that adults may show greater prefrontal cortex engagement, making threat safety discriminations, while adolescents might rely on earlier maturing subcortical structures. But to date, no studies have examined developmental differences in generalization of social versus non-social threat. So we wanted to examine whether developmental differences emerged during threat generalization and whether stimulus-related differences in threat generalization interacted with age. So our participants were an ethnically diverse sample of 16 adolescents and 20 adults recruited at the NIMH. And over the course of 4 visits, they completed 2 conditioning and extinction recall paradigms. One was a social threat paradigm and one was a non-social threat paradigm. So in visit 1 and visit 3, participants completed conditioning and extinction. During social threat conditioning, they saw pictures of 2 female faces. One of the faces, the CS plus, was paired with a fearful face and a loud scream. You might remember her. While the other face, the CS minus, was not. And in the non-social threat conditioning task, the conditioned stimuli were yellow and blue bells, and the CS plus was paired with the loud aversive alarm sound. So 3 weeks after each conditioning visit, at visits 2 and 4, participants underwent an in-scanner extinction recall task. So they viewed the CS minus, CS plus, and then an array of morphed images with different blends of the CS plus. And participants were asked 1 of 2 questions, either how afraid are you, which measures their threat appraisal, or how likely was this woman to scream or bell to ring, measuring their explicit memory of the conditioning event. So just to be clear, it's data from this visit that we're analyzing moving forward. So we used representational similarity analysis to measure pairwise dissimilarity in the patterns of neural activation elicited by each stimulus. So for example, you would take the correlation between the yellow bell and the green bell, take 1 minus that correlation, that gives you a dissimilarity value, and you input those into a dissimilarity matrix, and you get out of that 1 dissimilarity matrix for each brain area and subject for each task. So first we wanted to characterize the shape of the neural generalization gradients. And so for this, we were interested in how neural representations elicited by each generalization stimuli or morphed stimulus compared to the representation of the true threat stimulus, the CS plus. And so we ran a linear model with these comparisons, including age and task as factors, as well as linear and quadratic trends to characterize the shape of the generalization gradients. So here I've plotted the similarity of the neural patterns elicited by each of the generalization stimuli versus the CS plus, the true threat stimulus. The social task is plotted in red and the non-social task is plotted in blue. So we're comparing the similarity of neural patterns elicited by these generalization stimuli to the CS plus. So these are along the x-axis. So because we were interested in the shape of these gradients, I've plotted lines of best fit to just help visualize these shapes. We found an interaction between task and gradient pattern. So we found that social stimuli followed a linear generalization gradient. So here, for example, you can see it's linear. So whereas the non-social stimuli followed a quadratic generalization gradient across all regions. So meaning that in the social task, as stimuli increasingly resembled the threat stimulus, patterns of neural activation also resembled those elicited by the CS plus. On the other hand, in the non-social threat task, people showed increased similarity between the CS plus and the CS minus. So participants' neural activity during the non-social task reflected poor threat safety discrimination. We also ran a similar analysis on the behavioral ratings, and we found the same result where social stimuli elicited a linear generalization gradient across both threat appraisal and explicit memory conditions. And the non-social stimuli elicited a quadratic pattern of activation. So now that we've characterized the shape of the generalization gradients, we wanted to know how closely aligned are the neural representations with the subject's explicit memory of the conditioning event. So to compare the self-report and neural activity, we converted the explicit memory ratings into a dissimilarity matrix. So we've taken the absolute value of the difference between each score and put that into the matrix. And these matrices were averaged within each group and task. And we correlated the dissimilarity matrix for each region of interest to the appropriate behavior matrix via a Spearman correlation. And so this resulted in one brain behavior correlation for each subject in each task and region of interest. We then looked at the influence of age, task, and region of interest on behavior brain correlations using repeated measures ANOVA. And so here the brain behavioral correlations are plotted for the social and the non-social task for adults and youth. Social is plotted in red, non-social in blue, and adults are in the darker color and youth are in the lighter color. So first we found a main effect of task such that social stimuli elicit greater brain behavior correlations across all region of interests and age groups. We also observed a main effect of age, where adults demonstrated greater brain behavior correlations than youth. And finally, we see an age by task by region of interest interaction. So in the ventromedial prefrontal cortex, adults showed greater brain behavior correlations in the social task than the non-social task, but we didn't see this difference for adolescents. And by contrast, in the dorsal anterior singular cortex, adolescents but not adults showed greater brain behavior correlations in the social task compared to the non-social task. So we examined representations of social and non-social threats and generalization among adults and adolescents, and 3 key findings emerged from this study. So first, we saw that for both adults and adolescents, neural activity patterns followed a linear generalization gradient for social stimuli and a quadratic gradient for non-social stimuli. So the different patterns we observed across tasks suggest that socially relevant threat stimuli may elicit enhanced threat safety discriminations compared to non-social stimuli. And as mentioned earlier, this may be due to a need to have more precise recognition of social threat stimuli. Discrimination for socially relevant stimuli may be supported by humans' unique face processing abilities. So face processing occurs in specialized neural pathways, and faces are processed holistically, which may allow better discrimination between subtle differences in facial identity. So we also wanted to probe whether developmental differences emerged during threat generalization, and we observed a main effect on age on brain behavior correlations, with adults showing greater brain behavior correlations than youth. So one possible cause for this age difference is that adults may have greater access to autonomic threat detection processes than adolescents. So as interoceptive ability increases with age, it might allow adults to utilize physiological cues that result from activating threat relevant networks, which then they could use to inform explicit ratings of threat. Again, this design doesn't speak to this directly, so it's just a hypothesis. And then our third aim was to probe which brain regions perform differentially across social and non-social threat generalization, and whether this interacted with age. So we found that adults' brain behavior correlations to the VMPFC were greater for social stimuli than for non-social stimuli, but we didn't see these differences for adolescents. So the VMPFC is central in fear inhibition. During extinction recall, VMPFC activity increases as stimuli should resemble the safety cue. And so the VMPFC is still developing through adolescence, so adults may be better equipped to regulate autonomic threat responses to these safe stimuli or the CS+, CS-, to align better with their memory of the threat conditioning event. Alternatively, we found that adolescents' brain behavior correlations in the DACC were greater for social stimuli than for non-social stimuli, but we didn't see these differences for adults. So the DACC is involved in conscious appraisal of fear response, and there's some evidence for developmental differences in DACC response to threat-related stimuli. Adolescents showed increased DACC activation relative to adults when they viewed threat-related face stimuli, and this response was modulated by emotional expression. So while adults may engage the developed prefrontal cortex to make threat safety discriminations, adults seem to be relying on regions in the salience network to make such distinctions. And this could be important in studying aberrations in threat generalization, which are related to disorders like anxiety. Okay, so I think that's my time. So just want to thank my collaborators, Kalina Mihalska, Megan Peters, and Danny Pine, and the research teams at the NIMH and UC Riverside.
Great, thank you very much, Dana. And if you enjoyed the talk, please drop a note in the chat because we cannot applaud here on Zoom webinar. So thank you again, Dana, for a great talk. If you do have questions for Dana, please drop them in the Q&A box at the bottom of your screen. And as we're waiting for some questions to come in, because we have a couple minutes, Dana, I'll ask a question if that's okay. So it looked like in one of your plots there were negative brain behavior correlations. Do you think you could comment on that just a little bit? What does that mean to have a negative brain behavior correlation?
Yeah, so this I've been thinking about because I was initially kind of confused about this, and I don't have an answer for that. But what I think is happening is, since for these ratings we're comparing the whole dissimilarity matrix, and so for the first plots it was just versus the threat rating. Now we're comparing each threat stimulus to each other, and so looking at some of the neural dissimilarity matrices. I don't think I have them here. It looked like there may have been more differentiation for the CS minus. So I'm seeing more differentiation in some of the neural plots up here, which we're not seeing in the ratings. And so I think that might be driving kind of this like opposite effect where it's giving us a negative rating. So I'm not quite sure what to make of that. But I think that's maybe it's that there is more differentiation in neural patterns to the safety stimuli that is not represented in the behavior ratings.
Great. Things to look into in the future. Fantastic. Okay, well, if you have any remaining questions for Dana, please do drop them in the chat or in the Q&A, and we can answer in text-based format. So thanks again, Dana, for the great talk, and we will go ahead and move on to our final speaker for this session. So Alain, why don't you turn on your camera and share your screen? Great. And so this final talk in this session is from Alain Ferhat from the Zuckerman Institute at Columbia University. And he will be talking about reward-biased transitive inference task in rhesus macaques. And so it looks like you are ready. So why don't you take it away? Thanks.
Thank you. So hello, everyone. As I mentioned, I am Alain Ferhat, and I will present you our preliminary work on reward-based transitive inference task in rhesus macaque. So if I tell you that this kid A have better marks, better grades than this kid B, and B have better marks than C, you can automatically deduce or infer that A have better grades than C. Such deduction is called transitive inference and referred to the adaptative learning of novel relationship between adjacent pairs to mentally create a hierarchy. The learning process of this cognitive task can be explained using reinforcement learning. So, in basic way, we have two main models explaining reinforcement learning. The model-based, where the subject use experience to create an internal map, an internal model to response, and will follow the rules. And the model free, where the subject do not construct a model, but use experience in a simpler way to seek reward. And we can also have a kind of mixture of both, when model-based and model-free can co-opt or interfere. But all these two models interact during the TI task. So what happens when rules and reward interfere during our TI task? So to respond to it, we use a modified TI with dual list presentation. So this task consists to present pairs from two different lists. The first one, the pro list, contains 5 ordered items, A, B, C, D, E, rewarded by 2 draws. And the rich list, also containing 5 ordered items, but rewarded by 5 draws. We work with monkey, and before the task, the monkey have no expectation of these orders. So the pairs are randomly present during the task. If the monkey chooses the higher ranked item, he got the reward according to the list. Otherwise, he got nothing. So during the training, only within list pairs, adjacent pairs with a symbolic distance of 1 are presented. But during the tests, different other pairs will be presented according to the symbolic distance between items, distance of 1, 2, 3, or 4. Furthermore, items from with the same rank in each list can be switched. So this is the testing set. So at this point, we have four conditions. Concerning the within list presentation, we have adjacent pairs, the training sets, where, for example, A from the pool list is higher than B from the pool list with a distance of 1. And all possible pairs with A from the rich list is higher than D from the rich list with a distance of 3. But we have also the between list pairs presentation with adjacent pairs. In this case, there are no conflict because A is always the higher item compared to B, and rich is the more rewarded list compared to the poor list. So there are no reward conflicts. We also have all pairs presentation, but in this case, in this particular example, we have a conflict between A, the rank A higher than the rank D, and but D is more rewarded than the poor list. So we have, in this case, a reward conflict. So what do we observe concerning the behavior? First, for the within list pair, we observe the response accuracy, the performance in y-axis for each item A, B, B, C, C, D, D, E in x-axis for each monkey. In green, you have the performance when both item is from the poor list. In orange, when both item is from the rich list. And we observe similar performance with over chance level for both presentation, even with a reward disparity for the three monkey. Concerning the all pairs presentation, we observe the performance for each pair according to the distance of 1, 2, 3, or 4 between items, and we observe a slightly smaller performance for the poor list in green compared to the rich list in orange. We also observe an increase of performance according to the distance. Then concerning the between list presentation. Here the purple curve is when the higher item is from the poor list and the lower item presented is from the rich list. This list contain conflict between reward and rules. In pink, you have when the higher item is from the rich list and the lower item is from the poor list. And you have no conflict in this pairs presentation. So concerning adjacent pair in between list presentation, we observe high performance when higher item is from the rich list, and so a low performance for the list when items are in conflict. And for this list, we observe performance under the chance level. And this observation shows the influence of reward in the response ability. Concerning the all pair presentation, similar observation can be made for the purple for the conflict list, with an increase of performance according to the distance. So now we have some clue about the interference between rules and reward. And if we follow if the TI task follows structure census, the model based, the reward should have a very small impact. The performance should be similar for the both lists, and we should observe an effect of symbolic distance, the proof that the subject creates an internal map of the sequence. If the TI stars follow structure census model-free reinforcement learning, we should observe an important influence of the reward and preference for the wish lists for items from the wish list. But we still don't know the weight of rules and rewards on this particular TI task. So let's decompose the behavior into different parameters using regression analysis. So we admit that the performance is a function of the index of performance at trial 0, the intercept, but also the reward and symbolic distance have an influence of this performance. So first, concerning index of the performance in a within list. We observe the performance at trial 0, and for the within list, adjacent pair, and all pairs, we observe a positive intercept. So over the chance level for adjacent pair and all pairs. Then for the between list pairs, we also observe performance above chance level for adjacent pairs and a little smaller for all pairs. Then the second parameter is the effect of reward disparity. So here we can observe that the rewards have an influence around 0 for adjacent pairs and a positive effect for all pairs on the performance and on the learning. Concerning the between list pairs, we observe a much larger effect of the reward for adjacent pairs presentation and all pairs presentation, suggesting a large effect of reward thinking and a model-free reinforcement learning. Finally, the last parameters of our regression is the symbolic distance. And for the within list, but also for the between list, we observe a large effect of symbolic distance for all monkey on the performance during all pairs presentation. So this result suggests a creation of the mental map and the following of rules, and consequently suggest a model-based reinforcement learning. So to conclude, we observe that monkeys are able to perform TI tasks with a performance above chance for each task. This is the intercept, the performance at trial 0. We also observe a positive influence of symbolic distance on performance during all pairs presentation, suggesting a creation of a mental representation and a learning of the rules. And this result is really consistent with a model-based reinforcement learning. However, the reward have an important influence on the performance, especially for between lists, and this influence seems to be higher than the influence of distance. And this result is strongly consistent with a model-free reinforcement learning. So our results show that we cannot explain TI by only one model, but there can be an interference between rules, model-based, and the rewards of the model-free in the learning and the creation of hierarchy. So now we need to separate and quantify the impact of the both model and try to understand the mechanism behind. I want to thank my collaborators in Ferreira's lab, so Vincent, Greg, Albert, and Fabian, and thanks NIMH for the funding. Thank you for your listening.
Fantastic, thank you. And if you enjoyed the talk, please drop something nice in the chat because we don't have applause here in Zoom webinar. So thanks again, this is a great talk. If you guys have any questions for Alain, please use the Q&A box at the bottom of your screen and we can answer those as well. But while we are waiting for those questions to roll in, if they do roll in, ask your questions, guys. So I'm just going to ask a very quick question, just a clarification question. I noticed that there seemed to be kind of a precipitous dip around the pair CD in some of your performance measures. It was kind of earlier in the talk, and I was wondering if you have any thoughts for why pair CD would be particularly different than some of the other ones. A little bit, yeah, like here. So yeah, there it seems like, especially for the rich versus rich, but even also for some of the other ones, there's this kind of this dip in accuracy there, and I was wondering if you have any thoughts on that.
Yes, I have. I just have to
sure
to put myself in order. So here the A item is always rewarded and E item is never rewarded. So basically B, C, and CD should have similar performance. But what we observe usually in TI task, it's the CD pairs has always a lower performance. I've missed it's very frustrating. I'm sorry, I forgot the thing, but
it's probably some very simple reason that I'm not familiar with these tasks, and so I should know better. But we can also chat afterwards if you want. Does anybody else have any other questions for Alain? Anybody? Okay, well, you know how to find our speakers if you would like to follow up on any of these exciting results. Thank you again for the fantastic talk. And so again, you can find all of the speakers' contact information, or if you participate in the NeuroMatch matching algorithm, you might even get matched with them if this is your cup of tea. So I'd like to thank all of our speakers once again. If you guys want to turn on your cameras briefly and wave, say hi. If you're there, if not, that's okay. And thanks to the audience for being here, and we'll see you next time. Thank you. Bye. Oh, there's a question on YouTube. Hold on. Can someone, Gowrie, would you be able to copy that over? Okay, here we go. Is the positive coefficient of symbolic distance effect found sufficient to say the monkey follows a model-based learning somewhat? So, and then you can see that in the panelists chat.
So here we observe a positive effect of symbolic distance. It's how to respond to that. In just tech like this is not complete to respond to the question. So we admit that if the positive effect, if there was a positive effect, and we saw that in the performance with an increase of performance when the distance increase, we admit that the monkey can make the relation between A and D, for example, so with larger item, without learn the effective distance. So we admit that the monkey create a relation between AB, BC, and then CD to create. So it creates an internal model of the serial that we proposed. And this internal model follow the model-based learning.
Great. So I'm not sure who asked that question on YouTube, but we can go and find out if you'd like to follow up. And unfortunately, I have to cut this off now or we will get kicked out of the session for the next one. So thank you again very much, everybody, and we will see you in the next session. Bye-bye.